Hi, everyone. Thank you so much for being with us here today on, I guess, asynchronously on this recording. Thank you for all of your patience and your flexibility over the last couple of days. We know that it has not been easy for us and it has not been easy for you guys with figuring out what was going to happen with this session, but we're here. So hopefully this is good for you guys. Um,
Yeah,
yeah.
so just as like a recap, we did have a midterm review in person, I guess right before your midterm, and there's like a full thorough explanation of cycles 1 through 5. For this final session, we're going to go a little bit quicker like for pre-midterm content, just because we want to more heavily emphasize the later cycles. So if you need like more clarification on that, you can watch the midterm review, which is found on OWL.
As well, because this is asynchronous, we're going to be doing questions. So when we have a question come up, our mentor will read out the question and then they'll tell you to pause. And that's your cue to pause your screen, try the question on your own because we're going to go straight into going through the question or solving it rather than giving you the full minute in this video. But yeah, that's it. Introducing our first lovely mentor, Sarah.
Hi, everyone. My name's Sarah, and I'm going to be going through the first cycle, which is all about evolution is happening. So, next slide, please. All right, so you guys started off this cycle with an interesting case study all about HIV. And HIV stands for the human immunodeficiency virus. And this virus is what we would call a zoonotic disease, which is simply a disease that humans acquire from other species. So in this case, usually it's a non-human animal. And this occurs due to a spillover event, which is simply when a disease jumps a species barrier, and it's more likely to happen between species of like a similar type of physiological anatomy, and it's usually more harmful in the new species than it was in the previous species. Furthermore, you guys talked about HIV and how it was sequenced. So they took all the bases and they kind of determined it. And they realized that it's actually rather genetically similar to another virus called SIV, which stands for the simian immunodeficiency virus, which actually occurs in non-human primates, so like chimps, gorillas, etc. And speaking more about viruses, if you guys look on the tree of life, you won't actually find viruses on there just because we categorize them as non-living and they don't follow all of the seven characteristics of living organisms, which can be actually found in the speaker notes from your textbook section of the course. And a little bit more about viruses is you would categorize them as what we would call an obligate parasite, and they depend a lot on their host for survival, for food, for energy, and they use a lot of host machinery to kind of do this. And here we have an image of one of like a virus particle, and we can see the purple section is what we would call the lipid coat. It's not always found in viruses, but it can sometimes be found, and they're not the same thing as our phospholipid bilayer that we would find in our cells. And then we also have the genome, which can be either DNA or RNA based, and it can either be double stranded or single stranded. Next slide, please. All right, thank you. So let's talk a little bit about like how we treat HIV. And so there was this drug that was developed, and it's called AZT. And this drug specifically targets an enzyme called reverse transcriptase, so RT for short. And what it does is it transforms RNA to DNA, which is kind of different from what we do. We go from DNA to RNA to proteins, but this enzyme goes kind of the other way, and it does this so it can integrate its DNA into the host's DNA, which is very problematic. And so what happens here is when we're given this drug AZT, normally reverse transcriptase would be grabbing thymines and other bases to continue building its DNA. However, it grabs an AZT instead, and AZT is pretty similar in structure to thymine, but what it does is it stops the function of reverse transcriptase. So the reverse transcriptase isn't able to add more bases, which is very problematic because it's not able to continue its life cycle. But over time, what happened is that reverse transcriptase got a mutation where, it stopped picking up these AZT molecules, which made it very resistant to the AZT. Now, it's important to note here that the resistance does not occur because of the introduction of AZT, because mutations are random. They occur at any time. So it's important to note that this mutation did not occur because AZT was introduced. As you guys can see the picture there, you have the resistant form of reverse transcriptase as well as the original form. And then you guys kind of dove into another side of evolution, kind of just talking about theories in general. And scientific theories, they all need to be testable and they all need to be falsifiable. And if the theory is falsified, then you need to change your theory. And then this is completely different, but let's talk about evolution. So here are four core ideas of evolution. And the first one is that evolution is simply change in allele frequencies over time, as well as, all life all life in this universe is related through a common ancestor, which we denote as Luca. And then species change over time through like multiple generations, and mutations are heritable, and evolution is not predetermined, neither is it optimized for perfection. So there's no like end goal with evolution, and nothing really strives for that kind of perfection. Nothing's perfect. And then you touched on two, scientists. One was Lamarck, and you talked about his theory of evolution as well as Charles Darwin's. Lamarck's theory is incorrect. However, we still touch upon it, and you still need to know it for your final. So let's go through it. Lamarck's theory in simple is simply just change driven by individuals. So it's the idea of transformational evolution, that a species changes throughout its lifetime. So the common example given is with giraffes, and how a giraffe throughout its lifetime is able to stretch its neck long enough for it to be able to, reach trees and reach like the different leaves on the tree. However, that's incorrect. And that's where Darwin kind of proves him wrong, where he says that change is driven by natural selection as well as more like a variational approach, and it's driven by external pressures and how that change occurs across multiple generations. Next slide, please. All right, so this is our last slide, and this one simply just kind of falsifies a lot of the different statements about evolution. So the stuff with an X on it is kind of the misconception, and then the check mark kind of explains what it is in reality. So the first one is humans are directly descendants from chimps. This is like false. It's not true. We're related to chimps, so both humans and chimpanzees, we came from a common ancestor, and we both evolved to become different species. However, we're not descendants from chimps, so that's important to know. Another one is that humans are more highly evolved. This is false. No species is more highly evolved than another. We all evolved starting from the same time, and we all came from the same common ancestor, so we've been evolving for the same amount of time. Another point is that mutations are directed towards the needs of an organism. This is false because, again, mutations are random, and maybe they help an organism in a certain environment, but maybe they don't. So it's really important to know that, these mutations are random, but, sometimes they do help. And then the last point is that selection results in a perfect species. This is again incorrect because selection does not result in a specific type of perfection when it comes to a species. Rather, everything kind of has an evolutionary history. There's a lot of trade-offs to be made, and it's important to know that the environment's always changing. Maybe a specific mutation is helping, a specific species now, but let's say we change the environment, there's like a natural disaster, and now all of a sudden that is no longer advantageous. So everything's changing. So next slide, and let's do some practice questions. So I'm going to read this question, and then you guys can pause and take a minute to figure out what we think. So which of the following statements best explains the origin and characteristics of HIV in the context of zoonotic diseases and viral evolution? So pause and think about what it could be. Okay, I'm assuming you guys have thought it through. Next slide, Anna. So there we go. There's your answer. It's B. HIV is an obligate parasite that is likely originated from a spillover event. So these are really key terms. Obligate meaning, it's dependent on its host. Spillover event, remember, it's jumping the species barrier from one species to another. And it's a zoonotic disease. So it's, acquired from an animal. And the fact that it's genetically similar to SIV. And then the other ones are wrong. Well, first of all, for A is wrong because HIV is not living. Viruses are not considered living. That's the main reason why A is wrong. C is also wrong because, it's not directly from the human genome, and there is connection to other types of viruses when it comes to like SIV. Similarly, that's also why D is wrong as well, because they're not on the tree of life when it comes to classification. So they're not considered living. Next slide, please. Okay, here's the other question. Three flasks contain an equal number of bacteria reproducing at equal rates. Flask X is treated with an antibiotic ampicillin, and then flask Y is treated with two different antibiotic drugs, ampicillin and bacticillin, and then Z is not treated with any at all. So bacteria in which flask will develop antibiotic resistance through this mutation. Give you guys a second to think about that. Okay, I'm assuming you guys have thought about it. Next slide, Hannah. Okay, so the answer is A because all flasks are equally likely to get this type of resistance because mutations are so random that they could really happen in any of these flasks. So it's really important to know that mutations don't happen to kind of strive for a certain type of trait, but they just happen randomly. Let's do another one. Next slide. Okay, which of the following statements regarding evolution is true? Pause and try it out. Okay, next slide. All right, so D, humans can influence the evolution of other organisms. So again, this one is correct because, humans were constantly influencing the environment around us as well as nature around us. So we're always influencing, different types of changes and evolutions. Organisms, I will go through why they're wrong. A is wrong because organisms are not always getting better through evolution. There's no end goal, right? We're not always striving for something to get better. Sometimes, mutation does involve some species getting worse, and then through natural selection, you'll be, selected against. And then perhaps, that specific organism with that specific trait does not live anymore. And then B, evolution results in distinct species that can be clearly recognized. Sometimes it's really difficult to recognize these species. So that one is wrong. And then C, evolution anticipates future changes in the environment and allows organisms to adapt accordingly. No, that's not true either, because sometimes it is really hard to adapt to these different changes. And we don't anticipate any change because remember, there's no predetermined evolution. It just happens. So there's no control. And then I think we have one more question, if I'm not mistaken. Yes, okay. Which of the following traits is shared by all life as well as viruses? So pause and think about an answer. Okay, next slide. All right, so D, copies of DNA and RNA genomes are inherited by offspring. So this is important. So let's go through why each of them are wrong. A, genetic information is contained within a protein shell. So remember that the protein shell, that outer layer, isn't always there, only in some viruses. And B, genes are transcribed from an RNA code to a DNA message. So again, this only happens, the RNA to the DNA only happens in viruses through the enzyme called reverse transcriptase, not in humans. So only viruses. And then C, that one's wrong. DNA, RNA genomes are copied without error mutations. If that were true, then we would not have any evolution, we would not have any changes. So there's definitely errors and definitely mutations that occur, which is what helps drive evolution. And then that's it for my part. And I wish you guys all the best on your final exam.
All right. So thank you, Sarah. That was awesome going over cycle one. Now we're going to be moving into cycle two, which is all about the cell cycle and cell division. So this is more of the molecular nitty-gritties of biology itself. So yeah, let's just jump right into it. Next slide, please. Perfect. Okay, so first let's quickly go over the cell cycle. This is just the growth and what happens for each cell as it grows and divides and it, goes through its normal functioning. So first we'll start off with G1 phase, which is where the cell's really preparing to start dividing, where it's going through its normal functions, where it's preparing itself and building itself up to actually start replicating its DNA and then to hopefully divide eventually. And then we get to the next phase, which is our S phase, which is where DNA replication will occur. So this is where if you have those photos on our karyotypes where it's just like the one line of a chromosome. This is where it's going to have that X shape afterwards. So we're replicating that whole DNA. Then we move to our G2 phase, which is after DNA replication, and we're really preparing for mitosis, really preparing for the cell to divide. So the cell's growing, gathering all it needs for both cells to function as they can and by themselves. And then we get to our M phase or our mitosis phase, which is where cell division actually occurs. And this occurs in 5 steps. So the first is prophase, where the cell, the nucleus starts to dissolve, and we have the condensing of the chromatin into those chromosomes. The centrioles are starting to form those spindle fibers to connect to each of the chromosomes themselves. Then we have metaphase, where all of the chromosomes actually line up in the middle of the cell itself. Anaphase, each of the sister chromatids, so in the X shape, each of the sister chromatids are going to move to either side of the cell. Telophase is when the cells are actually starting to form themselves. So if it's one ball like this, then they start to branch off and peel off like that. And then cytokinesis is just that, cutting it in half to have two full final cells as themselves. And so that's the cell cycle. That's what it'll go through. Just one thing to note as well is that in G1, we can have another phase called G0, which is where the cell, instead of, it'll do its normal functions, but instead of preparing for cell division, it won't be preparing at all. So these kinds of cells won't be, prone to dividing. So you can think about like heart cells or like neurons. These cells won't really divide, and so they'll stay in G0 to, keep functioning as they normally do, but without dividing or preparing themselves for cell division. And so in our cell cycle, we actually have these three checkpoints here, which a cell has to pass or get through in order to actually go through cell division. If it doesn't pass a checkpoint, then the cell can be either arrested until problems with, maybe mutations or not enough nutrients or resources are fixed. And if those problems can't be fixed, then the cell will actually undergo apoptosis or programmed cell death. And so we have these three checkpoints here. The first one is in between our G1 and S phase. Our second is in between our G2 and our M phase, or mitosis, that's cell division. And then our third is the mitotic spindle fiber checkpoint, which is right, if you can look in here, squint your eyes a little bit, it'll be in this between the metaphase and anaphase. So right as the chromosomes are actually formed down the middle of the cell, if those spindle fibers don't form properly or like make correct connections, then the cell can be arrested at that point. Maybe the spindle fibers can be taken back and then reattached to the chromosomes correctly, or the cell can undergo apoptosis if it just all goes to hell in that moment. And then just one more point to point out for cell cycle and division specifically is, why do cells divide? And so they pretty much divide to maximize their surface area to volume ratio. So the volume of a cell actually will grow quicker than the surface area can compensate for. And so in order to, compensate for that, as the cell gets bigger, it'll break off into these two different cells. And once it does that, the volume of each is smaller, but the surface area is going to be enough to compensate, for that volume of each cell. And so this will maximize our surface area and volume ratio. The big thing to note for this is that you want your SA to V ratio to be high. That's one question that'll definitely, at least one question that'll appear probably on your exam is what the surface area to volume ratio is talking all about. The main point that you need to know is that that volume or that ratio should be high. And so let's move on to the next slide, please. Perfect. So at our checkpoints, we have these specific regulators that'll help either, progress a cell through those checkpoints, or it'll stop it where it is until, problems are fixed, or it'll undergo apoptosis. And so we can have these two different ones. We have our positive regulators and our negative regulators. Our positive regulators, intuitive naming, they're going to allow the cell to go through these checkpoints. And so these are going to be our cyclins and our CDKs. And all these are just proteins. And so they're individual proteins, but once they come together to form this complex here, as shown in the bottom, what'll happen is that cyclin CDK complex will actually phosphorylate another target protein, which will then go on to help the cell go through that checkpoint. So what you need to know is that cyclins and CDKs, if a cell can move through, what'll happen is those cyclins and CDKs will come to form one complex that'll phosphorylate another target protein, and that target protein can then push the cell through the checkpoints. And so these are how they're positive regulators. They get the cell actually through that checkpoint. If the cyclin and CDK don't actually bind together, then no phosphorylation can happen. The cell can't go through those checkpoints. And so that's how it can stop the cell cycle there. Moving on to our negative regulators, we have these three ones up here, our P53, which is our guardian of the genome, our P21, and RB. These are all different proteins, and these have, intuitive function as well with their name. They stop the cell or block the cell from going past checkpoints. And so they arrest the cell cycle. And what can happen when it's arrested is we can either, fix these different problems. So maybe a mutation's occurred in our DNA. We don't want that cell to divide, or else all other cells will have that genetic mutation, which could possibly harm their cells. So what'll happen is it'll either go back and fix those problems. So it'll fix that mutation and then continue the cell cycle. Or what can happen is apoptosis, as we talked about before. And so a lot of these, P53, P21, a lot of these are called tumor suppressing proteins because if we don't have anything there stopping the cell from progressing or from, dividing or proliferating unstopped, uncontrollably, that's the word I was looking for. If we have this uncontrollable growth, then that's where tumors will form. And so when these P53, P21, when they're deregulated, that's when cancer can form. And so that's why we call them tumor suppressor proteins because they stop those tumors from forming by stopping the cell cycle and stopping division. And so if we look down here, we can focus particularly on the action of P53. So what it'll be is that P53 will actually act as a transcription factor for the P21 protein over here. So P53 will bind to the promoter region and basically turn on the manufacturing or the production of P21. Then once we have a lot more P21 in the cell, that'll bind to our cyclin CDK, remember our positive regulator, that'll bind to that complex and it'll basically stop it from phosphorylating the cell or phosphorylating the target protein. And so that's how it can stop or negatively regulate the cell cycle in that sense. Next slide, please. All right, so let's quickly go over a practice question. So quickly, which of the following statements regarding the cell cycle and division is true? Just take a second, pause if you need to, jot down your answer in your mind, and then we'll take it up. All right, so hopefully you have your answer in mind. So let's go to the next slide and see the answer. Okay, so for this one, it's going to be C. The positive cell cycle checkpoint regulators are cyclins and CDKs. Obviously, we just talked about that. Those are what's going to push the cells through the checkpoints via phosphorylation. And so we can go through the other ones. A, the cell cycle consists of three phases, G1, S, and M. That's incorrect. We have four phases, G1, S, G2, and M. The cell cycle will continue to divide if it does not pass the checkpoints. This is the opposite, actually. The cell cycle will stop dividing if it doesn't pass the checkpoints. And then D, a cell cycle or a cell divides to decrease its SA to V ratio. Remember, this is incorrect. The SA to V ratio is always wanted to be high so that, we have enough stuff coming into the cell or the cell can actually, take in enough stuff to fulfill its duties and stuff based off of its volume. So that's why C is going to be correct in this scenario. Next slide, please. Perfect. All right, so we talked a bit about the cell cycle and cell division. Now let's focus in particularly on our gametes. So mitosis will occur in our somatic cells or our body cells, but our gametes specifically, or our sex cells, will undergo meiosis. And so there are two phases to meiosis. We have meiosis 1 and meiosis 2. The steps are pretty similar for both. The only difference is really in meiosis 1. What we'll see in meiosis 1 is that instead of those sister chromatids, so remember that X shape, instead of those separating, what we'll have is these homologous chromosome pairs. They'll line up at the middle line in metaphase, and instead of the sister chromatids separating, it'll be the homologous pairs themselves separating. And so they'll go to each side of the cell and go into their own cells as well and go into two new daughter cells. And then that will undergo another round of differentiation and, that division. And so here in meiosis 1, we call this reductional. So in this case, our ploidy getting reduced, and we'll talk about that in another second here. And so another thing that I want to point out for meiosis 1 is that this is where recombination or that crossing over of our non-sister chromatids occurs, which we'll talk about in a second. But then the other part, the second phase, meiosis 2, this is pretty much the exact same as cell division or mitosis. And so what will happen is those individual chromosomes will line up in the middle of the cell during metaphase. And what will happen is those sister chromatids will actually be separated in this phase. And so meiosis 2 is equational, not reductional, because there's no change in ploidy from when the first cells divide during meiosis 2. Next slide. Perfect. All right. So just want to point out a couple of differences in mitosis and meiosis. Mitosis is, as I said, only for our somatic cells or our body cells, whereas meiosis is only for our sex cells. And so the big difference between them is that in mitosis, there's no change in ploidy at all. So if you look in this picture on the left here, you see that in the parent cell, once the DNA replicates, we're still at 2N for our ploidy. And once those cells divide, we still have that 2N ploidy there. So there's no change in ploidy when we're undergoing mitosis. Whereas in meiosis, if we look at the top and we look at the bottom, we start at 2N, but then we end off with four daughter cells, each with N. So our ploidy is getting reduced by half when we undergo mitosis. And so that's mainly occurring in that first phase of meiosis, meiosis one. That's where those homologous pairs are actually going to separate to each side. And then meiosis two, those sister chromatids are going to segregate in that sense. And so the big thing is just meiosis is reductional and it occurs only in sex cells. Next slide, please. Perfect. All right. So, in meiosis, we these gametes that we make, there's a lot of genetic variation in them, and that's mainly because of these three reasons. The first one we've talked about already a little bit is that recombination or crossing over. And so that's going to be the exchange of genetic material between non-sister chromatids. And so this will happen in prophase one, where we have, once we have those sister chromosome or those homologous chromosomes line up beside each other, what will happen is the ends of the non-sister chromatids, if we look down here at the bottom left, the blue and the red chromosomes are actually, their chromatids are going to cross over and they're going to exchange that genetic information. And so that can lead to a lot of different genetic variation, a lot of, mixing of genes from one chromosome to another. And so this will contribute to some of that genetic variation in meiosis. One thing I want to point out and is important for this is going to be linked genes. So that's basically saying that genes that are closer to each other on the chromosome are more likely to be inherited together. So if we look down at this picture at the bottom here, gene A and B are more likely to be inherited together, whereas gene C is less likely to be inherited with either one of those. And so that's the only thing you really need to know about linked genes. Moving on to independent assortment, this helps with genetic variation because once we line up those chromosomes on the metaphase plate, what'll actually happen is if we look down here, we have these two bigger chromosomes in possibility one and these two smaller chromosomes in possibility two. What'll happen with independent assortment is that they'll line up on the middle plate or the metaphase plate independently of each other. So either possibility one or possibility two will happen equally. It's random whether they'll line up as possibility one or in number two, which way they'll orient themselves. And if you think about, we have 23 individual pairs of chromosomes, how much variation can happen in that with all of the different combinations of different, different types of chromosome pairings we can have in that. And so that's how independent assortment can influence our genetic variation. And then the last one is pretty intuitive, just random fertilization. A random egg cell and a random sperm cell are going to come together and form the zygote. And each of those are independent, they've gone through recombination, they've gone through independent assortment. So those two are very, very, genetically different from each other. And so that'll help contribute to much of this genetic variation, especially during meiosis and fertilization. Next slide, please. Perfect. So let's go over another question. Which of the following statements regarding meiosis is false? Just take a second to think about your answer, and let's move on to the actual answer. Next slide, please. Perfect. All right. So the answer for this one is going to be D. In meiosis 2, sister chromatids are separated into daughter cells. Remember that, meiosis 2 is what looks like similar to mitosis, where we're separating those sister chromatids, that X is getting separated. Going through the other answers, for A, recombination occurs during prophase 2. This occurs during prophase 1 when we still have those homologous chromosomes in the parent cell. And so that's where those non-sister chromatids will actually cross over in that sense. B, meiosis 1 is exactly like mitosis. It can't be exactly like mitosis. The big difference for meiosis 1 and mitosis is that meiosis 1 separates homologous chromosomes. Mitosis separates sister chromatids. So mitosis 2 would be more like mitosis, or meiosis 2 would be more like mitosis. So B would be incorrect. And then C, meiosis 1 is equational, meiosis 2 is reductional. It's just the flip of that. Meiosis 1 is where we separate those homologous chromosomes. Meiosis 2 is where we separate those sister chromatids. So yeah, let's keep going, move on to the next slide. Now let's get into a bit of this term that we call aneuploidy, and that's just basically saying that there's an abnormal number of chromosomes within the cell. So if you think of, Down syndrome, that's a trisomy 21. We have three chromosomes at that chromosome number 21. And so that would be called aneuploidy, just the abnormal number. When we're usually supposed to have two, they actually have three. And so this can work for any different combinations, whether you have one chromosome, three, four. Usually we wouldn't really have four, so we only talk about three and one. But anything that really isn't normal or has an abnormal amount of um, chromosomes in the cell is going to be termed aneuploidy. And so for our purposes, let's really focus in on during meiosis. This can happen via non-disjunction, and that's basically just saying that there's a mix-up or like an error in the separation of those chromosomes in the cell itself. One thing to point out is that most autosomal trisomies are lethal. So a lot of, if we have trisomies or extra chromosomes in our body cells, then that can be lethal or be fatal to an individual. But with sex chromosomes, aneuploidies can be viable. So if we have, two X's and a Y or three X's, or just a singular X chromosome, those can still be viable. A person can still live with those, non-disjunctions or separations. And so we can have non-disjunction in two different parts during meiosis. So we have those two rounds of separation or division, right? Now we can have non-disjunction in either of those points. So during meiosis 1, what will happen is instead of both of those homologous chromosomes, those homologous pairs separating to either side, what will happen is both of them will go to one side of the cell, and then the other cell won't have anything. It'll just be zip. So we can see in this bottom example here on the bottom left that these two red chromosomes, those are the homologous pairs, those are both being pulled up towards the top part there. And so once the cell separates, all of the resulting cells are going to have aneuploidy. So these top two cells are going to have three chromosomes. These bottom two are going to have one. And so that's the big difference for meiosis 1 is that all of the cells are affected in some way and all of them have aneuploidy. During meiosis 2, what will happen is, those homologous pairs are actually separated normally. But when we go through that second round of division, what will happen is those sister chromatids aren't actually separated properly. What will happen is they'll go towards the same side of the cell. And we can see this in this middle part during meiosis 2 on the bottom right. These two red sister chromatids are moving towards the same cell. And so what will happen is we'll have two normal cells. So they'll be, normal functioning. They'll only have two chromosomes in the bottom part there. And then these top two are going to be aneuploidy. So this top one that, got both of those sister chromatids is going to have three chromosomes, whereas the bottom one is only going to have one because that one's the one that's separated normally. These red chromosomes are the one that's separated differently. And so for meiosis 2, only some of the gametes are going to be aneuploidy. Only some of them are going to be different than what we normally see. Next slide, please. Perfect. And so the last thing that we can touch on is just the life cycles. Now, I'm going to be honest, I was like all over the place with this last year. It's definitely a bit confusing when you first try to look at it or reason these out. But as long as you can follow a few principles with these, it's easier to understand the mechanisms of each. So if we look at the animal, what we normally talk about for humans and what we intuitively know, we have two phases. We have the diploid phase and the haploid phase. The diploid phase is when we're going to be only in our two ends. So that's going to be when, starting from the sperm and the egg, they come together and they make our zygote. That's going to be diploid. Then that separates or divides by mitosis to create a mature being or a mature animal. So say myself, I've a product of mitosis from a zygote. So are you, so is everybody else. And so what'll happen is we're all in that diploid phase. We're all in diploids, but once our gametes start being, produced, what'll happen is those will undergo mitosis, and our gametes are the only part of, our body or our animal's life cycle in which they're in this haploid state. And so then the sperm and the egg, those gametes will come together, those haploid cells will fertilize each other and become the zygote at the left side over here, and that cycle will go round and round. Moving on to the other two, we have similar, mechanisms in each of them. So for both the land plants and the other fungi and algae, they both have diploid and haploid phases. The main difference is only just in the timing of how long they're a haploid, how long they're a diploid. And so the big thing, the most complicated one is going to be these land plants because we have the sporophyte and the gametophyte. One thing that I like to remember for the land plants compared to the animals is that their diploid phase, when they're in two end, is pretty much the exact same. So if we look here, both of them are made from zygotes. Both of those zygotes divide and grow via mitosis. And then we have this mature kind of diploid organism here. In animals, it's going to be, the human, the actual organism itself. In plants, it's going to be the sporophyte. So that's just that mature diploid organism. And then they undergo meiosis afterwards to create haploid organisms or haploid cells. And so for our land plants, what'll happen is instead of making gametes right away, they'll make what are these spores down here. And so these spores can replicate themselves. It's pretty much just a regular cell. So you can think of like the zygotes. The spore is just going to be like a haploid zygote, pretty much. It doesn't need fertilization, but it's going to be able to grow and replicate on its own and divide. And so this spore will undergo mitosis, divide into this gametophyte here. And so that itself is, its own entity. So it has pretty much like two phases of being mature, the sporophyte and this gametophyte. And then from the gametophyte, instead of undergoing meiosis to make gametes, what'll happen is it'll just divide via mitosis to create the gametes. Those will be fertilized like normal, and then we'll get the zygote and go around in the cycle again. And so that's really the most complicated part is just this gametophyte and the sporophyte, which is, diploid, haploid. The way I like to remember it is just focusing on the gametophyte. So gametophyte sounds, that has the same starting as gametes. So I know that that's going to be haploid. And uh, because the land plants divide differently, what'll happen is I just remember that the gametophyte separates itself into those gametes. So it'll undergo mitosis into those gametes. And so that's how the fertilization occurs. And then for the sporophyte, that'll undergo meiosis to create its spore. So the gametophyte make the gametes via mitosis, and the sporophyte makes the spores via meiosis. It's kind of a bit counterintuitive to what you'd think. Gametes being made from mitosis, what the heck? But as long as you can make that distinction, it'll make answering questions on the exam about these much, much easier. And so the last part is just the fungi and algae. This one's pretty much the exact same in our haploid phase to our land plants. The only difference is this diploid phase, and all that happens is there's no real mature diploid organism for algae or anything. Once the gametes fertilize each other, they become the zygote, and that zygote undergoes meiosis right away to create the spores. And so the spores will divide via mitosis to create the gametophytes, and then the gametes are made, and then fertilization. So the zygote and this diploid phase is very, very short. Once the zygote's made, it just undergoes meiosis right away into the spore. And so as long as you can understand, how these different cycles are related to each other and, which phase is diploid, which phase is haploid, and then what kind of mitosis division happens at each point, you should be good. So once you start thinking about these and practicing questions, it'll be easier to understand these relationships. And so let's do one practice question for that real quick. Next slide, please. Perfect. So which of the following differentiates the animal life cycle from the plant life cycle? So take a second, pause the video, read those questions out, and then jot your answer down in your head. Okay, So hopefully you have your answer now. Let's move on to the answer. So for this one, it's going to be A. Animal gametes are produced via meiosis. Plant gametes are produced via mitosis. So remember, animal gametes come from, our cells. We make our gametes or sex cells from meiosis. The plant's gametes are going to come from that gametophyte. And so that gametophyte's just going to split off into two different or these different gametes, and then those are going to be fertilized by each other. So Plant gametes are by mitosis, animal gametes are by meiosis. For these other ones, question B or answer B, the zygote immediately undergoes meiosis in plants. Um, no, this one's incorrect. The zygote immediately undergoes meiosis in algae and fu most fungi, but in plants themselves, the zygote will, differentiate and separate into that sporophyte before undergoing meiosis. And so that's what you need to know for plants. Question C, zygote matures to animal via mitosis. Plants mature to sporophytes via meiosis. This one's incorrect. Remember that diploid phase, that maturity of the zygote, the growing of the zygote is pretty much the exact same in both animals and plants. So as the zygote grows, it'll divide via mitosis for both plants and animals to create the mature organism. And then D, the haploid phases are the same, but the diploid phases are different. Um, no, this one's just incorrect. Animal and plant cell haploid and diploid phases are different. While the diploid phases may seem similar, the haploid phases are much, much different. The plants have that gametophyte, that mature haploid organism. The animal life cycle only has those gametes that come together. So no, D would be incorrect. Perfect. So that's all for me. Good luck on your exam, guys, and hope you guys knock it out of the park.
Okay, hi everyone. So I'm going to take you through cycle three, arguably the best cycle, but Hannah, I'm going to have you proceed forward. Okay, so this is just a quick summary slide of everything we're going to be going through. I find it really good when you're studying to just like condense everything onto one page, but I'm going to break this all down, NNC value, karyotypes, and then DNA replication. So next slide, please. Okay, so the big question, why do we care in the first place? It's really important because the genome reflects what's happening in the cell. To understand what's going on with an organism, let's start at the basics. Let's look at the genome. We also want to look between species, so understanding different genomes, how do they equal different organisms? And then, of course, evolution of genomes, looking at the past and the present, and leaving us to talk about complexity of certain things. But please remember, for the rest of your discussions, you need to keep in the forefront of your mind, complexity is arbitrary and dependent on definition. So the genome size might not necessarily reflect complexity of an organism depending on the context you're defining it in. So next slide, please. Okay, everyone's favorite topic. So NNC values, they're really peculiar because N focuses on a really nifty rule, but we're going to break it down and you're totally going to understand it. So N value is defined as the number of unique nuclear chromosomes in an organism. So you have your coefficient that is going to be your ploidy, and then that's going to illustrate the number of sets. So another way to think of this is your complete chromosome count. When you're talking about your complete chromosome count, please remember that you count chromosomes by the centromere, not by chromatids. C value, on the other hand, represents the amount of DNA in one set of an organism's nuclear chromosomes. This is your genome size. And then, of course, your coefficient tells us how many times that set is present, but this is the actual amount of DNA. So looking at the bottom graphs here, we can see, okay, along the cell cycle in each of them, when we're looking at N values and the coefficient of N, we have in a human, it stays 2 the entire time. Whereas if we're looking at the coefficient of C in humans, it doubles. So remember, when we are talking about this, there's a really in-depth explanation on the notes of this slide as well, if this doesn't suffice, but hopefully it does. When we're talking about this, because we're talking about the centromeres, when you go through the cell cycle and you replicate the DNA, since N value is defined as the number of unique nuclear chromosomes, when you replicate, for the sake of this course, you're going to assume that those chromatids are identical. So those are no longer considered, that's not an extra set, that's not unique. You're just replicating something you already have. So coefficient stays the exact same all the way throughout. When you separate, you still have the same amount of chromosomes you did at the start. You still have the same amount of centromeres. You can either think about it as, let me just count the centimeters, or let me recognize the fact that this new replicated set is not unique to the original one. In terms of C value, this is a lot easier to understand because when you replicate, you are physically doubling the amount of DNA you have there. If someone hands you an apple and then hands you another apple, you're going to say, okay, well, this one weighed half of what both of them weighs together. That's exactly what's happening. If you were to take all the DNA during G1, put it on a scale, you're going to get a given number. If you were to take all the DNA after it's been replicated, put it on a scale during G2, it's going to double. That's exactly what C value is going to tell you in the terms of you have a set, and then the coefficient is going to reflect whether or not it's increased. Okay, next slide, please, Hannah. All right, practice questions. So this practice question actually has 5 parts, just kept it concise, all based off of this diagram. So what's the ploidy of this organism? What's the end value? For number 3, please don't get confused about the letter C. That just means coefficient. And then that's going to reflect like the total chromosomes in the genome based off the image provided, how much DNA is shown, and base pairs. So please use the C value in the bottom corner. And then if the cell were to replicate, how would it affect the N and C value? So give it a quick pause, ponder it, and then come back. Okay, hopefully you pause and you come back now. Next slide, please, Hannah. Okay, so breaking this down, we can see right off the bat, this is a diploid organism. We have 2 sets, right? We see blue, yellow, pink. Great, we have 2 sets of each. N value, again, we're thinking unique. Well, even though we have 2 sets of each, we only see blue, yellow, pink as unique, distinctive chromosomes. There you go, that's your 3. And then putting that together, we know diploid, that's going to be our coefficient, multiplied by 3 equals 6. Now we're talking about C value, so we know that C was the weight of one set of the chromosomes, but we have 2 sets of these chromosomes in this state. So 3.7 times 2, 7.4. And now this is the tricky part. You're thinking about if the cell were to replicate, how would it affect the N and C value? Okay, N is not changing. N and C value themselves, the actual value of each of them is not changing. The only thing that would change would hypothetically be coefficients. The coefficient of N, not changing. Nothing's happening to the coefficient of N. The coefficient of C, however, is going to double. That's because during replication, you're doubling the DNA. Coefficient of N, not changing because the number of centrometers stays the same. Okay, next slide, please. All right, so that was all the fun stuff. Now it gets a little bit easier. Karyotypes are basically just us taking a picture of the genome. So it's taken during metaphase. Everything is paired up. Everything has been replicated. Super nice. On the side here, I made it a point to put in 2 karyotypes. In the top one, you see your characteristic, like hallmark chromosome looking type, like the X's, right? Great. And the bottom one, that is still a human karyotype, only it looks a little peculiar because you can't see that like X characteristic. It just looks like 1 chromatid, 2 chromatid. The one on the bottom is something you're more likely to see on an exam, which you probably did on your midterm, but please recognize the fact that in both karyotypes, in the one in the top and the one in the bottom, there are 2 chromatids in each chromosome. So you have 4 in the top one, right? You see your 1, 2, 3, 4 if we're looking at chromosome 1. And at the bottom one, the chromatids are just so close together you can't see them, but there actually is like 1, 2, 3, 4. But it looks like 1 chromatid. So just make note of that. Karyotypes can tell us the ploidy, right? How many sets do we have? It does not tell us C value, right? We're not weighing these things out. We can't actually count how many base pairs are in this state. So that doesn't tell us any information. But unique chromosomes talk about the number chromosome number, not how many you have of that number. So again, that's just like a logistical thing. Certain definitions you're going to have to memorize, but luckily with karyotypes, it's kind of easier to just intuitively look at them and interpret what's going on there. And then, of course, remember, all the alleles might be different. So you might have like a heterozygote, right? You have 1 allele on 1 chromosome, 1 allele on another, but the genes are the same. Okay, next slide, please, Hannah. Okay, so super quick practice question. Pause, give it a read over, let me know what you think. This one luckily has a nice diagram with it, so if you're struggling, don't worry, but give yourself some time to go through it. Okay, that should be enough time. Perfect. Okay, so this is the visual solution for that karyotype. The answer to the previous question was, in fact, A. And I took the liberty of drawing it all out because I know it's a lot to kind of think about all in your head. But when you're going through it, the statements, you need to be really careful about whether or not it's stating chromatids or chromosomes. So for example, statement 1 says the karyotype will show 90 chromatids, right? And that's distinctive because remember that karyotype I showed you guys actually has 2 chromatids in each of those chromosomes, right? So this would be reflective of that karyotype on the bottom of that slide, 3 slides back. So when we do the math, ends up being 90. Second statement said karyotype analysis would have been right before cytokinesis. We know that's false. It happens at metaphase. You still have to go through anaphase, telophase before you get to cytokinesis. Third statement said the weight of the entire genome, all 5 copies, would equate to 12 million base pairs, which we know is true, right? Because if the C value for one set of the chromosomes is 1.2, and then we have 5 sets of the chromosomes, that gives us 6. But remember, this is during metaphase, so that doubles again, giving us 12 million base pairs. Great. And then last statement says karyotype reflects a change from 5n to 10n. No, we just established that the coefficient of n is not changing in this case. Great. Next slide, please. Okay, now let's unpack DNA replication. So this is the really fun stuff. We know that these are just some of the basics. You have your purines, pyrimidines, your base pairing, what bonds. So I'll leave you to read over that. That's not something that's super challenging. But please remember some certain facts about DNA. You have anti-parallel double helices. So what that means is basically one strand will read 3 to 5, that ends there, and then you have your 5 to 3 going up, and then it wraps around each other. Super cool. DNA is semi-conservative, which basically means it separates, and then one of the original strands serves as a template. One of the original strands serves as a template, and then it forms 2 new helices together. So one is an older strand, and then one is a newer strand, or parent strand, new strand. And then DNA polymerase reads 5' to 3', and then synthesizes, reads 3' to 5', synthesizes 5' to 3'. I could never remember this in first year. The way I like to remember this is, okay, we read left to right. When you write out the numbers 1, 2, 3, 4, 5, 3 comes before 5, that is left to right. DNA polymerase also reads 3 to 5, left to right. So that's a little tidbit. Hopefully that helps you out there. And then, of course, you have your leading and lagging. That's simply because of the way the replication bubble opens up. You need to lay a primer in order for things to be synthesized, but since the primer placement is dependent on when you can actually access that DNA, you need to wait for it to unwind, unravel, open up, and place it. So we'll get into that in a second. That's how you end up with those Okazaki fragments and then one continuous. Okay, and then please note that you can have multiple bubbled in eukaryotes and one in prokaryotes. Prokaryotes are pretty efficient with it, but anyhow, next slide, please. Okay, so unfortunately, these are enzymes you're just going to have to remember, but they're pretty intuitive. So helicase unwinds your DNA, right? You're taking it apart at the ends. Topoisomerase, this is going to relax your hypercoiled DNA. And what this means is think about an elastic. Think about, you're a kid, you're winding up a plain elastic and it gets really, really tight. And if it's like wrapped around your finger, you can feel like the pressure like cutting off circulation on your finger. What topoisomerase does is at certain points, it would kind of nick it to relieve that pressure on whatever's getting coiled down the line. Because if you were to start opening your fingers like that, everything that you've been twisting here is going to feel a lot more tense. You need to relieve that. That'll damage your DNA. That's exactly what topoisomerase does. Then you have your primase, that's laying your primers. Then you have your DNA polymerase 1, removing the primers, filling gaps. DNA polymerase 2, synthesizing strands. And then your ligase to mend the gaps between the primers and the fragments of DNA. Okay, next slide, please. Great. So we're going to take a couple minutes or a couple seconds, I guess, to do a practice question. So pause your screen or the video, I guess, and then give this a read over and then come back and we'll take it up. Okay, should be good. Oh, there should have gone. Okay, it's okay. There should be another one, but it's fine. The answer is A. So let's break this down. We'll start from D and work our way up. So the question basically says, hey, this was on a test back in November, and this diagram was actually flawed. What's wrong with it? So D says there should be Okazaki fragments on both the top and the bottom strand. Well, we know that in the case where we are unraveling DNA and we are dependent on placing primers when the DNA is accessible, one of them is going to be able to move smoothly, right? Because we have to still follow the rules of 3' to 5' in terms of reading and 5' to 3' in terms of synthesizing. In this case, that means one of the primers will have a nice continuous strand and the other ones will have those little tiny ones that need to be glued together. By saying Okazaki fragments should be both on the top and bottom kind of makes that statement null and void. So we know D right away, wrong. C says the blue strands should be labeled new strands instead of parent strands. Okay, so if we were to label the blue strands as the new strands instead of the parent strands, we would be saying that that is the DNA that we are newly synthesizing. That's incorrect because that's the DNA that's actually unwinding and acting as the template for our newly synthesized strands. B says neither should have a lagging strand since the replication bubble is opening up from the end of the chromosome. The primer is able to be laid in an unhindered way. Now, this seems like a really enticing answer, but please remember the fact that you are still needing to abide by the 5' to 3' synthesis rule. Those strands are anti-parallel. They're oriented in such a way where the primers are still dependent on being placed in a certain region such that DNA polymerase can synthesize in the 5' to 3' region. If the strands were parallel, you would be able to do this, but they're anti-parallel, meaning on what you have on one strand, you can't do on the other. So it's reversed on both. That leads us to A. The leading strand synthesis should be towards the fork, and the lagging strand synthesis should be away from the fork, which makes perfect sense. Every diagram we've seen has shown leading strand towards the fork. Again, because it is abiding by that 5' to 3' rule, and it just so happens it can do that in the direction that the fork opens on both sides. So if that still doesn't make sense, don't worry. There's another explanation on this one as well, but let's keep going forward. Okay, so this is the really fun stuff. Telomeres. So you could think to yourself, okay, DNA is replicating, that's great. When you get to the end of the chromosome, to be able to synthesize new DNA, you're contingent on having a place to put your primer. When you reach the very, very end, if I have my chromosome here and I want to synthesize this DNA, but I have to put a primer here and there's nothing there, you can't put it on anything. So a certain region of DNA is going to get lost. If that happens every single time a cell divides and this DNA was important, let's say this codes for something really important in your body, that wouldn't be really good. So our bodies have a mechanism to combat that. We have telomeres. So that basically is just a region of repeating sequences on the end of eukaryotic chromosomes that protect this important DNA. So it would extend further. So if we're going to lose anything, it's just going to be this buffer region that we're like, yeah, that's disposable. That's okay. But remember, each time we replicate, you're still losing something. So every time the cell replicates, you're picking away at those bases on the end. And every time it replicates, that's another replication down. And the amount of times the cell can replicate before reaching that, precious DNA is known as the Hayflick limit. And once you get to the Hayflick limit, you guys learned about cell senescence. It's basically where the cell cycle arrests. Now, please don't think that cell senescence is equated to the cell dying. It's not in any sense. Cell senescence, you can think of it as retiring. Like it can still do its work on the side, but it's not doing anything crazy. Like it's not going through the cell cycle, it's not replicating, it's minding its business, it's doing its job, but it can't go through the cell cycle any further. Because if it were to go through the cell cycle any further, it would continue to replicate, and then you'd start losing the important DNA. As you start losing the important DNA, you create opportunities for things to go wrong because now you're omitting this DNA, the cell's dividing, it's giving rise to new cells that are lacking really important genetic information. And then that lack of genetic information can give rise to things like cancer because it would be interpreted as a mutation and things can go awry. So we actually have this really cool enzyme in all of our cells, but it's not expressed in all of them. Telomerase restores the length of the chromosome. So it can actually go back and add some buffer region on. Telomerase, if we had it in every single one of our cell types, could be really problematic. Even though in theory, it's like, okay, this is great. Like I could live for a billion years if I wanted to, if telomerase was just hanging around. But no, on the other hand, telomerase would just lead to uncontrolled cell growth. You'd just be adding things wherever you wanted. Cells would just be dividing constantly all the time. And telomerase is actually a hallmark sign of cancer. Cancer cells express telomerase because cancer cells are like, they're literally fighting for their lives. Like they just want to grow and do whatever they want whenever they want to. Telomerase is also expressed in stem cells, which makes sense because those stem cells need to proliferate. They need to have that proliferative capacity and they need to be replicated with fidelity without having any worry of losing that important DNA. And then lastly, the RNA primer synthesized in the newly extended template strand is within the telomerase enzyme. So the telomerase enzyme will come in, you have your RNA template strand, you're still placing everything, and then you'll have your extended region. So in this case, telomerase is something you do need to have a relatively good understanding about. Do you need to know all the nitty-gritty mechanisms? No. If you have questions about them, feel free to reach out, but hopefully that makes sense. And I'll be handing it off to Jason for cycle 4.
Okay, thank you so much, Selena. So cycle 4 is a bit of a short cycle. We only had one lecture in this cycle, and we're going to start off by talking a bit about DNA mutations and repair. So we're going to start off quickly with a bit of a primer question. So why do we care about this? Well, a lot of the reasons have already been brought up by Hannah and Matt. For example, when we look at DNA mutations, they are a major driver of genotypes for evolution, as well as mutations that cause disease, like a lot of different types of cancer. You'll also talk probably about cystic fibrosis in the next semester, as well as a lot of other diseases. For example, Hutchinson-Gilford, Paget syndrome is something we're learning a lot about in upper year. All right, so next slide, please, Hannah. So we're going to start off by talking a bit about where do these mutations come from. Then we're going to talk about what mutations are. And then finally, we're going to end with how to repair these mutations. So just quickly, we have two major sources of mutations. On the left, we have exogenous sources. Or sorry, I've actually flipped these. Endogenous sources on the left. And sorry, we're gonna look on the right side first here. Endogenous sources on the right here. So DNA replication error, mitochondria will produce reactive oxygen species, as well as spontaneous tautomerization. The last of which we will talk a bit more about later. I've got a lot of questions about mitochondria and the production of reactive oxygen species. So essentially what happens here, just briefly, is that at the end of your electron transport chain, which helps you make ATP, your last molecule that receives the electron is an oxygen. And sometimes this doesn't work very well and it produces what we call an oxide radical. And it's really, really unstable and it wants electrons. So what it'll do is it'll go over to DNA and grab some random electrons, and then that'll make the DNA unstable, and then that could cause damage to the DNA. And then in the middle diagram here, we have the exogenous sources. So we're looking at radiation from the sun, for example, X-rays, cigarette smoke, barbecuing, and of course, some viruses. Next slide, please. All right, so let's talk quickly about the types of DNA mutation and go over some really important key terms here. So we're going to start off with the point mutation. Essentially what this is that in one spot in our genomic DNA, we have a single base pair changed, altered or deleted. So next we have a transversion here. So if we look at our graph on the right side here, so a transversion, I like to think it's a bit of a longer word, so it's a bit more complicated. So in this case, we're going to go from either a pyrimidine to a purine or vice versa. So purines are on the top. So these are our nucleotide bases with 2 rings. And then our pyrimidines are on the bottom, and these only have 1 ring. And then a transition mutation is just we're going from a pyrimidine to another pyrimidine or another purine to another purine. 2 other key terms that we really need to distinguish is DNA mutations and DNA damage. So DNA damage is change in the original sequence on only 1 strand, and then a mutation is on both strands. And I have an example coming up in the next slide. Hannah, please. Thank you. All right. So we can start off here with just our normal DNA sequence. You can see we have our 2 strands. And then if we incorporate a DNA damaging agent here, which causes a mutation, as we've highlighted with C, we can see we have DNA damage. This is only on 1 strand. We've only changed something on this top strand here. However, when the cell divides, that's when DNA mutation happens. So on the bottom here, our strand is completely fine. When we do DNA replication, both the strands are pretty much separated. And in those 2 areas that Selena was talking about, that information is isolated. So in the bottom strand, we don't have access to the top strand's information. And in the top strand, we don't have access to the bottom strand's information. The bottom strand is doing fine. But if we look at what happens in the top strand, we have a mutation now. All right. So now, what are the types of mutations? Next slide, please. So we'll take a deep dive here. Let's start off with the substitution, which is essentially just a point mutation here. And as I've mentioned before, that is a change in 1 base. And then we'll look at deletions, which you can see we have 2 nucleotide base pairs in the sequence deleted. An insertion is, of course, just the opposite. We're adding something. And then an inversion, what's happening is essentially we're taking a middle piece of that strand and then we're rotating it 180 degrees. Now let's take a quick peek at the different types of point mutations. So when we look at this, we want to keep the central dogma in mind. So just to briefly reiterate, the central dogma is us going from DNA to RNA and then RNA to protein. So among all those things, DNA and protein are the most important because they are the most stable. And with protein, that's what actually carries out the function. So if we look back at past cycles, for example, Matt was talking about P53, as well as the RB or retinoblastoma protein that are really important for keeping that cell in check to stop cancer. Those are proteins. And those are the things that are carrying out the important functions in the cell. So let's start off by looking at the DNA level here. When we have no mutation, we go to mRNA and then we go to protein. Our amino acid in the protein is lysine. This is normal. In the second option here, a silent point mutation, this is kind of in the name here. We're not seeing a difference in the protein level. And that's because the codon for lysine, there are multiple codons that share this kind of redundancy for lysine. And so we don't actually see that at the protein level. Our AAG codon in the mRNA codes for lysine, and so does the AAA. Now let's look at our second type of point mutation, the nonsense mutation. Essentially what's happening is our codon is getting changed to the stop codon. One little swap from an A into a T, a T into an A here. And then when we go into the mRNA here, we get a U, we get a stop codon. And then that's going to completely stop the translation of our protein. Now, if this happens kind of earlier in the transcript, that's going to be a massive issue. You're going to be missing half or even more of the protein. Then lastly, on the right, we have our missense mutations. So we have two types of missense mutations. We're going to go over the conservative missense mutations. Essentially, what this means is we're swapping from one amino acid to another amino acid. So at the DNA level, you can see our second base in this codon, which is T, is getting switched into a C. Instead of coding for lysine, we are now coding for arginine. Arginine, the amino acid, shares a lot of characteristics with lysine. If we just look at the shapes, you can see they're kind of both really, really long. There's a nitrogen at the end of it, and they both have a positive charge. So if this happens, the protein can likely retain some of its function because, let's look at the amino acid. It's pretty similar. There's not too big of a change. So some of the function of the protein is going to stay. And then we look at a non-conservative missense mutation. So instead of a positively charged amino acid that has a nitrogen at the end of it, we have something with a hydroxyl group and a methyl group, threonine here. So in this case, because these two amino acids are so different, we're probably not going to get the similar function in the protein. And if this happens in some really, really important parts of the protein, this can be really, really bad. Okay, next slide, please. So now we're going to talk briefly about tautomers. So tautomerization is a shift in preferential base pairing, and it's a spontaneous event. So you guys have probably learned about isomerization a little bit in chemistry. This is kind of like that. It's kind of like a little bit of an isomerization, just a small, tiny change. However, this does not result in a mismatch, but can cause a mismatch after replication, which is a really, really important concept. So to quickly bring this up, we have our table down here, and we have our normal base pairing and then our tautomerized base pairing on the right. So in our normal base pairing, we can see the T is with an A. Now, if the T tautomerizes, it will naturally wants to bind to a G. However, if replication doesn't happen, the cell just stays like that. The T is tautomerized. It's still paired with an A. That's not a mismatch. We still just have T and A together, right? Nothing is wrong. However, when we tear those two strands in our DNA apart and replicate and then undergo our cell division, one of the cells is going to be left with a T to G because in its tautomeric form, our T wants to base pair with a G. And one really easy way to remember how tautomers base pair is just to think about purines and pyrimidines. So because in this picture here, this is our DNA double helix structure, we can see at the bottom it has a 2 nanometer width. So this is actually really important that we keep this width throughout the DNA to maintain its structure. And one of the ways we do it is that we always keep a pyrimidine bound to or base pair to a purine. So 2 rings base paired with 1 ring. And that makes things a whole lot easier. So if T naturally wants to bind with A, its tautomer has to base pair with G because G is our other purine. Okay, I think that's pretty much it for tautomers. Next slide, please. So another way to kind of introduce errors in our DNA replication is going to be our DNA polymerase slippage. So it can do two things. It can slip forward and backwards. Now, DNA replication is actually very, very good at being very accurate. So these kind of things are rare, but they do happen. Next slide, please. So, sorry, Hannah, again. So when it comes to a forward slippage, what we're getting is actually going to be a deletion in the synthesized strand. So you're going to want to think about how if we skip a piece of DNA, if we're skipping forward, then we're going to be losing a part in our template strand. We're going to be skipping that part, right? So in that sense, we're going to be deleting that part that's skipped. And if that part that's skipped continues to keep going, or sorry, the synthesized strand continues to be synthesized and it keeps base pairing with whatever is forward, then we're going to have a little loop here that's highlighted that's kind of going to be hanging out in our template strand, unbase pairing, because we skipped that part. We don't have that part in the synthesized strand. And when we talk about a backward slippage, it's going to be essentially the same concept. When we do a backward slippage, essentially we kind of copy the same part twice. So we'll go through this CAG region, CAG, and then we'll want to copy that again. And then our synthesized strand is going to keep going, but now we have this huge extra bit in our synthesized strand that we copy too many times. And it's got nowhere to base pair with. So it's going to hang out and it's going to have this loop. Next slide, please. So now that we've gone over all these mechanisms that cause DNA damage and mutations, how do we fix these things? Because you and I, everybody lives a pretty healthy life. Not everybody's getting cancer. We're exposed to the sun all the time. So the cell actually has a lot of mechanisms to handle these issues. One of these mechanisms is through proofreading as well as mismatch repair. So both of these actually fix mismatches, which is when two bases have incorrect base pairing. And this is often caused by a polymerase replication error. So you want to think a mismatch repair is almost kind of like essentially DNA damage. It's this change in one strand, and we're getting a mismatch between our normal, or sorry, we're getting a mismatch that is different from our normal base pairing. So in this example on the right, you can see we have a T base paired with a G, which is not right. So one of these mechanisms, as I previously mentioned, is proofreading. So essentially the DNA polymerase will be chugging along. It'll realize, hey, I put in the wrong base. And then it'll go back and use this 3' to 5' exonuclease activity. And it'll chop off that last nucleotide that it put in. It'll remove that. It'll fix it, and then it'll keep going again. And then when it comes to mismatch repair, we do this with endonuclease activity. So if we look at the diagram on the right side, we can see the red arrows in the first picture at the top here. This is showing endonuclease activity, which is essentially a few enzymes, which are kind of like molecular scissors. They're coming in, they're cutting inside the area where we have our mismatch. They remove an entire section. Our DNA polymerase will come back in. It'll synthesize that section, and then ligase will come back in and then seal up that little nick that DNA polymerase 3 left behind. Now, one quick thing to briefly mention is the difference between exonuclease and endonuclease. So an exonuclease is going to be on, you're going to want to think exo, outside. It's going to be on the ends of the DNA. So when you're synthesizing DNA with polymerase, and it removes that last nucleotide that's at the end of the new strand. Endonuclease cuts inside the DNA, kind of like you see in these red arrows. So next slide, please, Hannah. So our second way of fixing things is through non-homologous end joining. And this only happens when we have double strand breaks. So when double strand breaks happen, which can be caused by reactive oxygen species or ionization from radiation, the cell kind of goes into panic mode. It will literally do anything and everything to fix itself. Because if it doesn't, it's going to die. And non-homologous end joining is one of these mechanisms because the cell is so desperate to fix itself. We are going to do anything, even if it introduces errors. So essentially, the cell goes in. This is really, really sloppy, and it slams together both ends. It can add nucleotides. It can remove nucleotides. It can invert them, do all sorts of things. And we should note that, again, there's no template when this happens because these chromosomes are kind of just floating out and about in our nucleus. Okay, next, please. So to kind of wrap up the cycle, we want to talk quickly about transposable elements. Barbara McClintock, who did a lot of pioneering work on cell biology, discovered these. She helped with a lot of things like centromeres and karyotyping. So when it comes to transposable elements, they're appropriately named jumping genes because they're little sections in our genomes that like to jump around and move around. It can essentially insert itself as well as it can, again, kind of cut itself out, move around, and then reinsert itself. This is one of the major reasons where we have really, really large genomes is because of these things and a bunch of other factors. So these can be really, really problematic when we have issues like in the next slide. Like this, for example, if these genes actually these transposable elements insert themselves into a gene or into an area that controls a gene or cause the shuffling of a gene. So on the left side, we have factor 8, which is part of our clotting cascade. So our L1 transposable element is just a transposable element that we use as an example. If it jumps in the middle of this gene, and then we want to make protein from this gene, our transcript and everything is going to be completely messed up because we have a bunch of gibberish from our transposable elements here. And because now this protein doesn't work, we're going to result in a hemophilia phenotype. We can also have insertions that lead to gene shuffling. So essentially when a transposable element goes into gene 1 here, and then it's removed, it doesn't just take out itself. It takes out this entire part of gene 1. And then when it's added, it is now added together with that gene to a new section. And because the expression of a gene, the amount of transcript we make and the amount of protein we make is all regulated by this promoter, as we can see here, PR1 or PR2. And different genes have different promoters that regulate them. This can really, really mess up how much gene 1 product we're getting. If promoter 1 is expressed really, really low, it's a really weak promoter, we're getting a tiny bit of gene 1 normally. However, now that gene 1 is in control of promoter, is in the control of promoter 2, we can make a lot of gene 1. And this can be really, really bad if it's a protein that drives, for example, cancer or disease. All right, so now we're going to move on to our question. So we have 2 questions here. So cytosine can spontaneously undergo deamination, converting it to uracil in DNA. During DNA replication, uracil pairs with adenine instead of guanine. If a cytosine in a single DNA strand undergoes deamination to uracil after the first round of replication, but before the second round, what is the ratio of mutated cells to wild-type cells among the 4 daughter cells after our full 2 rounds of replication? So I'll give you guys a minute to go over this question. Okay. So I assume you guys are all good. The correct answer for this question is going to be C. So we'll briefly go over it here really quickly. If you guys are watching the recording, you have the slideshow. The answer for the question is going to be in the speaker's notes. So when we undergo our first round of replication, we're going to go from 1 to 2 cells. Now, both of these cells are going to be normal because, as the question says, we're only undergoing deamination after our first round of replication. Because of that, one of these cells is always going to be okay. It's because the deamination happens only in one of these cells. So that one okay cell will pretty much divide again in our second round of replication and give us 2 daughter cells. So in this entire scenario, we are guaranteed to have 2 normal wild-type cells no matter what. That essentially removes option A from contention. Now we're going to want to think about this other cell that undergoes that deamination process. So essentially what happens when that occurs is that one strand, say the top strand, has a cytosine being converted to a uracil. Now, uracil doesn't normally base pair with a guanine. Uracil wants to base pair with an adenine. So when it undergoes that second round of replication, as we saw with kind of that diagram in the previous few slides, one of the daughter cells will have a uracil that is base paired to a adenine. And then the other cell will have a normal cytosine to guanine double strand. So essentially, we're going to get 1 out of our total 4 cells that's mutated. Pretty much exactly like that example that I've shown you previously. So the correct answer here is going to be C. We're going to get 3 wild-type cells and 1 mutant cell. Okay, so on to our next question here again. We'll give you a quick minute to read the question. And yeah. So now that you've digested the question, the answer to this question is going to be B. So let's quickly go over the answers and see which ones are correct and which ones are incorrect. So during DNA replication, DNA polymerase slippage occurs at a repeat sequence. What factors is the most critical in determining whether this results in a nonsense, missense, or silent mutation? So this is a lot of different things that we have to think about. So we're looking at DNA slippage, but the question is also asking about nonsense, missense, or silent mutations. So what about DNA can cause missense, silent mutations? That's going to be a change in our base pairs and a change in the codons at the end of the day. So if we look through our options here, the length of our repeat sequence is not going to be correct because changing the length is going to change the length of our overall protein, but it's not going to cause one of these point mutations. The specific codon affected by the slippage is going to change it. C, so that's going to be the correct answer. C, whether the slippage occurs in the leading or lagging strand, which is kind of part of that replication bubble that Selena was talking about, that's also not going to be something that would cause a missense uh, or nonsense or silent mutation because that's again going to change the length of the repeat sequence. And then the exonuclease proofreading activity of the DNA polymerase isn't going to cause any mutations at all as well because that's again fixing the mutations. Okay, thank you.
Thank you, Jason. So in cycle 5, we're going to be covering Mendelian inheritance. In Mendelian inheritance, Gregor Mendel reached 4 important conclusions with his experiments. So the following rules are the basics of Mendelian genetics that we're going to cover. Very importantly, Mendelian genetics does not apply to all traits, and we're going to be covering some traits that actually don't follow this rule. So be careful when you're answering questions that you first determine whether you should use these rules or not. So the 4 rules are variation in traits is due to alleles. Alleles divide into gametes at random. Organisms inherit 2 alleles per trait. What's important to note about the third rule is that this is actually not true for most traits that humans have, because we have lots of polygenic traits which show continuous variation. For example, skin color is determined by lots of alleles, and as you know, there are lots of skin colors. 4, appearance of heterozygotes is determined by dominant alleles. Next slide, please. So I think the most important skill in this cycle is learning how to do genotype charts. So very quickly, alleles, they're the different variations of a gene. And then we're going to be covering the 3 types of genotype charts you're likely to cover. The first are monohybrid crosses. So they're covering 2 individuals who are heterozygous for 1 gene. You can see this in the bottom left. These crosses should be 2 by 2. And as you can see, you just assign each row or each column to have 1 letter. Pretty simple. The next type are dihybrid crosses. So you cross 2 individuals who are heterozygous for 2 genes. As you can see, it's the middle chart, and each row or column contains 2 letters, and they contain every possible combination. So your dihybrid cross should contain a 4 by 4 cross. Lastly, and very importantly, are sex-linked crosses. So these are used for when the gene is either found on the X or Y chromosome. And very importantly with these types of questions is that the sex of the individual can often determine the likelihood of inheriting a disease. For example, let's consider the chart on the bottom right with X and XH. If we consider XH as a allele causing a disorder, you can see that there is a 50% chance that a son could be affected by the disease, but 0% of daughters would actually have the disease. You would only have a 50% chance of having a carrier daughter. Next slide, please. So a really quick and useful formula for determining how many possible unique gametes can be formed is 2 to the power of n, where n is the number of heterozygous pairs in the genome. So we're going to be quickly covering an example of this. So the genome of an organism is given below. How many distinct gametes can be produced? I'll assume you tried it out. So the answer for this one is 16 because there are 4 distinct heterozygous pairs. You might notice that there are 5, but big B is not heterozygous. Next slide, please. So now we're going to be quickly covering 2 examples of non-Mendelian inheritance. So these don't follow the rules we outlined before. So the first one is incomplete dominance. So the heterozygote leads to an intermediate expression between the 2 alleles. And the second one is codominant, where the heterozygote expresses both alleles, but only quite slightly. It can be quite difficult to remember which one is which because they can sound quite similar. So I like to remember these examples through flowers. So in the example, let's say big A produces a red pigment and little a produces yellow pigment. An incomplete dominance, what would happen is that you would just produce an orange flower, just one color. However, if you have a co-dominant trait, you would have a red and yellow flower, but no orange. Next slide, please. The next example of non-Mendelian inheritance is epistasis. So it is a form of non-Mendelian inheritance in which one gene can interfere with the expression of another gene. A great example of this is, let's say A again codes for red flowers and little a codes for yellow flowers. However, there's another set of alleles. Big B allows for pigment to be deposited, but little b does not allow pigment to be deposited. Big A and big B are both dominant. So as you can see on the bottom left, let's say we have three variations, all of which have little b, little b. You can tell that all of these variations will actually produce the same phenotype of flower. This is because with little b, you don't have the ability to actually deposit any pigment. So no matter what pigment you actually form, it could be red, yellow, like orange, because you have little b, none of the pigment will be deposited and you'll end up with a white flower. So now we're going to be covering some questions to test your understanding. So in question, I'll let you read the question, but for this question, I would highly recommend making a genotype chart. It'll be really helpful. So the answer for this question is A. The answer is A because if you form the chart, what you'll see is that every flower receives a big A allele. And most importantly, in this question, it asks you, what is the expected phenotype of the flowers under Mendelian genetics? So you expect to use the four rules we outlined at the beginning, which says that the phenotype of a trait is given by the dominant allele. The question states, in and under Mendelian genetics, the capital letter is typically dominant. So you would expect all four flowers to be purple. You should also expect only four because this is only one trait. There's not two. Next question, please. So the second question asks you for a sex-related disorder. I would also highly encourage you to form a genotype chart for this question as they'll be really helpful. So I'm assuming you'll have paused and had an answer. The correct answer for this question is C. This is because the affected mother has both copies of affected alleles, and we know that the disorder is recessive. This means that all of the offspring will receive an affected allele. However, what's important is that because daughters have two X chromosomes, this means that each daughter will only have one affected chromosome and one normal chromosome from the father. This means that all of the daughters will be carriers. So 100% of the daughters will be carriers. However, 100% of the sons will only have one affected X chromosome, which means that all of the sons will be affected. This is because they are XY instead of XX. Next question, please. In question 3, I would take some time. So the answer to this question is D, for only. I'll cover all the statements. So the first statement is that it is governed by Mendelian genetics. This is false. This is because under Mendelian genetics, you would not expect to see this distribution of traits. And also under Mendelian genetics, you would expect that each trait is only governed by one set of alleles. And in this, you can see that the dog's color is there are three distinct dog colors, which you would not expect. Statement number 2, the first 16 offspring of these parents will always be 9 black dogs, 4 yellow dogs, and 3 brown dogs. What's very important to understand about genotype charts is that they're not exact predictions of, oh, these are the first offspring you'll get. They're just the relative odds of what is likely to be the genotype and phenotype of the offspring you produce. You could have a scenario where you produce double dominant homozygote like 3 times in a row. That could happen just because of probability. The third statement, the brown dogs are an example of incomplete dominance. This is false. This is because with incomplete dominance, you would expect to see all heterozygotes, and you would not also expect to see this type of distribution, phenotype versus genotype. The fourth statement, this is true. This is because if you look at the chart a bit more carefully, you'll notice that every yellow dog has little e or homozygote for little e. And this suggests that the little e allele is probably recessive for something to do with pigment deposition. And so the correct answer is D. And with that, that is the end of cycle 5 and the end of pre-midterm content. And I will hand it over to Hannah for cycle 6.
Hi everyone, I am taking over for cycle 6. Before I get started, I just wanted to say it's been a long hour and a half. So if anyone has been sitting here this whole time, please feel free, go grab a snack, go grab some water. You can pause us right now. We'll be fine. Definitely take your break. Like you can give your mind a rest. Okay, so getting started with cycle 6. So the first thing we're going to talk about is genotype frequencies versus allele frequencies, because I find that this is a topic that people tend to get a little bit confused by, and it is a really important topic for later calculating Hardy-Weinberg equilibrium. So genotype frequency is your distribution of your genotype in a population. So that means you're looking at like the actual genetic makeup of a trait compared to the allele frequency, which is looking at the distribution of a specific allele in the population. So I like to think of like alleles versus genes, kind of like baking a cake. So your gene is like your recipe, and then the allele is like the specific flavor. And that kind of like helps me clear things up. So let's look through and sorry, let's look through an example. And then hopefully that will make some sense. So in this above example, we have 120 red flowers, 10 pink flowers, and 70 white flowers. So that means we have 20 individuals total, or our population size is 20. And what that means is that we have 400 alleles total, because for each genotype, you have 2 alleles. So first, we're going to calculate genotype frequency, though. So to calculate the genotype frequency, it's as simple as we have our homozygous dominant, which is big A, so that's 120 and 120 red flowers, it says, and that's you divide that by the total population, and that gives you 0.6. Then we would do the same for our heterozygous genotype. So that is 10 pink flowers divided by the total population of 200, and that is 0.05. And then homozygous recessive, we have our 70 white flowers, or 70 little a, and over our population size of 200, and that is 0.35. Allele frequency is a little bit trickier, but it's not too bad once you get the hang of it. So the first thing we're going to do for allele frequency, so we're going to start with our homozygous, or sorry, with our dominant allele, so our big A allele. So the frequency of our big A allele, we are taking 120 times 2 big A alleles from the homozygous dominant. Why? Because the homozygous dominant has 2 copies of this big A allele. And we're adding the 10 copies of this dominant allele from the pink, which is the heterozygote. And then we're dividing that by our total number of alleles, which is 400. And that's giving us our allele frequency. Then for the allele frequency of big A, we're taking our 70 white flowers, which is again our homozygous recessive, and we're multiplying that by 2 because the homozygous has 2 copies of it. And then we're adding on our 10 recessive alleles from the heterozygote, and we're dividing that by our total number of alleles, which is 400. And again, that gives us our number of 0.375. So Hardy-Weinberg equilibrium. That's kind of what this was all leading up to. So Hardy-Weinberg equilibrium describes a set of conditions where there are no mutations, all genotypes are equally fit, close population, no gene flow or migration, infinite population size, random mating, and no selection. Now that's kind of like a long just checklist of things. So the way that they'll usually tend to apply this would be to say, like, is it possible that there's selection happening in this population? Because you would know that under Hardy-Weinberg equilibrium, there is no selection. So then you would calculate if the population's under Hardy-Weinberg equilibrium. And if it is under Hardy-Weinberg equilibrium, that means that there's no possibility that there is yeah, there is no possibility that there's selection occurring. But if it isn't under Hardy-Weinberg equilibrium, then selection could be occurring. So sorry. Um, Okay, so yeah, allele frequencies are not changing. The population is not evolving. There's no selection. That's the main thing. And really, there's just these two formulas that you need to know. So our first formula is p squared plus 2pq plus q squared equals 1. And p squared is the frequency of our homozygous dominant genotype. 2pq is our frequency of our heterozygous genotype. And q squared is the frequency of our homozygous recessive genotype. Now, it doesn't really matter which one you determine to be P or Q, but that's how they do it. That's how they taught it in your lecture. And honestly, it helps to keep things straight if you just always keep it the same way. And then P plus Q equals 1, where P is the frequency of our dominant allele and Q is the frequency of our recessive allele. So let's run through a quick example together. So if we have allele T associated with the ability to taste a particular chemical, and that is dominant over allele T associated with the inability to taste it, after surveying 100 people, 36 people can taste the chemical and 64 are non-tasters. The tasting gene was sequenced from all the people who can taste the chemical, and the results show that 24 of the tasters are homozygous and 12 are heterozygous. Is this population in Hardy-Weinberg equilibrium? So I will give you guys a second to pause it if you'd like to try it on your own right now. Okay, I'm going to assume you all paused it. We're going to go through it. So firstly, I highlighted some things within this question and I just wrote them down here. So population is important. So our population is 100 people. And then it also tells us the genotype frequencies of our population. So the genotype frequency of our homozygous dominant is 24%, of our heterozygote is 12%, and of our homozygous recessive is 64%. And because our population is 100, we can multiply that by 2 because there's 2 alleles per person in the population, right? So that means our number of alleles is 200. And the first thing that we're going to do to calculate if this is in Hardy-Weinberg equilibrium is find the allele frequency. So that's what we just did, right? So we're finding the allele frequency of the dominant allele. So finding the allele frequency of big T, it means we take 2. So we have our 24 out of 100 or 24%. So we take 24 times 2 from the homozygote dominant plus 12 from the heterozygote over 200, which is our total number of alleles. And then that gives us the frequency of 0.3, and that is equal to P, right? Because P is the frequency of our homozygous, or of our dominant allele, sorry. And then because we know that P plus Q equals 1, we can say Q equals 1 minus P, which is equal to 1 minus 0.3. Sorry, I should have spelled it out. So Q equals 1 minus 0.3, which equals 0.7, and that is our frequency of our recessive allele. And then now that we have the allele frequency, we can calculate the genotype frequency in Hardy-Weinberg. So in Hardy-Weinberg, the frequency of our homozygous dominant is going to be equal to p squared. So that is 0.7 squared, and that is 0.49. Then the frequency of our heterozygote is 2pq, or 0.42. And then the frequency of our homozygote recessive is q squared, or 0.3 squared, which is equal to 0.03. Now, we can just convert these to percentages really quickly and compare that to the population genotype frequency. And I can tell right away that those are not the same. So even if you didn't do it on your own before, can you say now, do we think this population is in Hardy-Weinberg equilibrium? The answer is no, because the genotype frequencies do not match the population, or the Hardy-Weinberg equilibrium genotype frequencies that we just calculated do not match the population genotype frequencies. That means that this population is not in Hardy-Weinberg equilibrium. Okay, moving on. Fitness and selection. So absolute fitness is the average number of surviving offspring for each genotype. Relative fitness, or the little w, so absolute fitness is a big W, relative fitness is a little w, and that is the absolute fitness of one genotype divided by the absolute genotype of the most fit genotype. So that's W max, which is our maximum fitness and the most successful genotype. And you might just have like a quick question that would ask you to calculate that on an exam. So you should know this formula. So what happens when we are selecting for an allele? So that's the definition of evolution, right? But first, what happens if all alleles are equally fit? So if all alleles are equally fit, Will the recessive allele disappear or not disappear? Can solidify your answer. I had this as if we were going to be raising hands in NCB. But yeah, the answer is it does not disappear. That's because it's not under any selection. So the recessive allele wouldn't have any pressure to disappear. But moving on, so if we had selection against which, a dominant or a recessive allele, which one would remove all copies from the population? I'll give you a second. Yeah, so selection against a dominant allele removes all copies from the population. So that is shown here where we have our fitnesses, right? So if the fitness of the dominant allele is less than the fitness of the, um, actually it should be equal here. I'm going to change it, but okay. It should be equal to the fitness of the heterozygote, and great less than the fitness of the homozygote recessive. And that would mean the dominant allele frequency would decrease to zero. So selection against the heterozygote. And then selection against which would cause the alleles to never fully disappear. Yeah, so selection against the recessive allele, the allele will never actually fully disappear. So what happens here is that the recessive allele hides out in the heterozygote. And that means that even though we're selecting against the recessive allele, so the genotype that's just recessive or the phenotype that's just recessive, the issue is we can't fully select out the recessive allele because it still exists within the heterozygote. So what about when there's a distinct heterozygote? So our heterozygote now has its own distinct phenotype. Um, an example of this could be an incomplete dominance or codominance. So which one, selection against the heterozygote or the homozygote, which one would cause the alleles to balance at 0.5 frequency? Yeah, so the answer was heterozygote. So selection against the heterozygote causes alleles to balance at 0.5 frequency because we are that means we're selecting for the homozygotes and we're equally selecting for the homozygotes. And that maintains genetic variation. This is an example of selection without evolution because at this point where they balanced out, so where they're both at 0.5, technically we're still selecting against the heterozygote, which means so we're still selecting against the heterozygote, which means there is selection present, but there is no evolution present because the frequency of alleles is not changing, and that's the definition of evolution. Okay, so I think you can probably guess, but selection against which, the heterozygote or the homozygote, the rare allele disappears from the population. Yeah, the answer is the homozygote. So this is heterozygote disadvantage, and what happens here is whichever allele starts off as more rare will disappear from the population. And this is one of the few cases where we can remove the recessive allele. Okay, so this is just a quick table to summarize it. I'm not going to go through it now. You can go through it on your own time. Let's go through non-random rating. So, sorry, non-random mating. So our mating types, assortative mating is referring to your, so sorry. Assortative mating is when you're mating with like. So you're selecting a like trait for a like trait at one trait. So you want to mate with someone who has the same trait of yourself. An example of that would be a ginger-only dating app, for example. So say you're a ginger, you'll only date other gingers. That is an example of assortative mating at one trait, and that trait would be the ginger trait. Now, population-wide or genome-wide, sorry, not pop, yeah, genome-wide, that is inbreeding. So that means you're selecting for people who have the similar genome as a whole, not just for one trait. And both of these things increase homozygosity. Now, dissociative mating is that one trait again, but that's when you're looking for traits that are opposite for yourself. So maybe you have brown hair, but you want to date someone who has blonde hair, and that is dissociative mating at one trait. And then inbreeding avoidance, which is just the opposite of inbreeding, which would be like within a family. So we're not mating with our family members. That is genome wide. So that means you're avoiding within the whole genome, and that increases heterozygosity. So non-random mating does not change allele frequencies, it just changes genotype frequencies, and there is no evolution necessarily occurring because evolution means the changing of allele frequencies. Now, genetic drift is kind of just an aside, but So genetic drift is random sampling error. So it is always happening in a population. I know that often we're taught about it as like a bottleneck or the founder effect. And yeah, those are really good examples of genetic drift, but you're properly emphasized that this is something that's always happening in the population. And pretty much it just causes allele frequencies to change over generations due to chance. Okay, we are going to go into our first question. You guys can stop here, pause the video, solve it yourself. Okay, gonna assume you all did that. And the answer to that is B. So why is the, yeah, so why is the answer B? Because this says foxes have difficulty seeing dark or white rabbits. Foxes that have difficulty seeing dark or white rabbits are introduced to the population and become the main predator. So what changes do we expect in this population over evolutionary time? So in this example, we are selecting against the heterozygotes. That is heterozygote disadvantage or homozygote advantage. And if you can recall from the table or from what we just went through, homozygote advantage causes the rare allele to decrease to zero. So because the rare allele in this case is going to be the recessive allele, and you can tell that kind of just intuitively. Like if you wanted to calculate this, you definitely could. But for me, I could just see, well, there's 16 homozygote recessive and there's 36 homozygote dominant. So the recessive allele is rare. So because we're under a state of homozygote advantage, that's going to cause the rare allele or the recessive allele to decrease to zero. Yeah. Okay, next question. So consider two alleles, L1 and L2, at the L locus controlling whisker length in a population of mice. There are millions of mice in this population, so we can ignore the effects of genetic drift. The starting frequencies of alleles L1 and L2 are 0.8 and 0.2 respectively, under which of the following scenarios will L1 eventually reach a frequency of 1 and L2 eventually disappear completely. So again, pause it, try to solve it yourself. Okay, so the answer to this is A, or 1, 2, and 3. So we're looking for which of the following scenarios L1 will reach a frequency of 1 and L2 will disappear completely. So 1, the fitness, this is saying that the fitness of L1, or the homozygote, is greater than the fitness of the heterozygote, which is greater than the fitness of L2, or the other homozygote. So at this point, I would say that's probably correct, but we also don't know for sure because we don't know. Actually, no, we do know for sure. Yeah, that is correct because that has the greatest fitness of all of them. And because it's greater than the heterozygote, it means that it's going to select for this, and the frequency of L2 will disappear completely. Now, question 3 is the same. It's the same reason. It's just a simple advantage. Question 2 is a little bit different. Question 2, again, we're pointing to this heterozygote disadvantage or homozygote advantage. So again, it's causing you to see that L2 is the rare allele. And because it is the rare allele, this will decrease under conditions of homozygote advantage. So yeah, 1, 2, and 3 are correct. 3 is correct because it's the same as 1. It's just that the heterozygote and the homozygote L2 have the same fitness. But overall, it doesn't change anything because L1 still has that greater fitness. Okay, one more question. Populations that are highly inbred, that is where individuals often mate with their close relatives, often have high levels of harmful genetic disorders. Why? Okay, I will give you a second. So the answer is D. Inbreeding increases the frequency of homozygous genotypes, and this includes genotypes that are homozygous for recessive alleles. So why are the others wrong? Well, A and B are talking about increasing the frequency of alleles, and as we said, selective mating doesn't, or non-random mating doesn't change the frequency of alleles. It just changes the frequency of genotypes. And then C, inbreeding decreases the overall mutation of rate in the population, allowing harmful alleles to accumulate. Again, it's not decreasing the mutation rate. Doesn't and decreasing the overall mutation. This answer just doesn't really make any sense, and it's not related. Um, this question asks you to go through Hardy-Weinberg 4 times. I'm not going to go through it now, but it is really great if you're struggling with like kind of getting the basics of Hardy-Weinberg, which we already went through. Would recommend going through it. The answers in the speaker notes. And that is it for me. Thank you.
All right, hi guys, my name's Roshan, and I'm going to be going over cycle 7, which is about sexual reproduction. So before I get into the actual lecture content, I just wanted to talk about a big overarching theme for this whole cycle, which is that sexual reproduction is useful because it increases the genetic diversity of offspring. This allows for the emergence of genotypes with higher fitness or better fitness for changing environments. And then if we contrast this to asexual reproduction, or just popping out identical clones, what we find is that since sexual reproduction gives us diverse offspring, these offspring can have greater fitness in the parents, or they can have greater fitness. They can have fitness that allows them to adapt to changing environments. Whereas with asexual reproduction, it's just the same exact genome and fitness, same genome every single generation. So it's not fitness isn't going to decrease necessarily, and it might not might not be able to respond to changing environments. So this idea of increased genetic diversity of offspring is what they're referring to whenever they say sexual reproduction allows for new combinations of alleles. Next slide. Okay. Oh yeah. So just some terms for you guys to know. This is kind of just raw memorization, but yeah. So to categorize sexual reproduction, first we can categorize them into isogamous species, which are species that have equal sized gametes. This is in contrast to anisogamous species, which are species with gametes that are either small and mobile or large and non-mobile, with the most obvious example being human sperm versus eggs. Human sperm are small and mobile, and their eggs are large and non-mobile. So with isogamous, you can more or less just think of it as all the gametes between all the individuals look the same. Whereas with anisogamous, you have sperm, eggs, they're super distinct from each other. The only other thing to know for isogamous species is that they have this concept of mating types. So they may have 0 or multiple mating types. And essentially, the example she gave in lecture was that yeast gametes can either be an A mating type or an alpha mating type. And what we find is that only some types can mate with each other. So with yeast, A's can only mate with alphas, but A's can't mate with other A's. That's all there is to know for isogamous species. Then moving on to anisogamous, these 4 terms at the bottom here, monoecious, sequential, simultaneous, and dioecious, these are subcategories of anisogamous. So anisogamous implies that there's 2 sexes. So these organisms can either be monoecious, aka hermaphrodites, where each individual has both male and female function. Then at the bottom there, you have dioecious, where each individual is one of 2 possible sexes, like with humans, male and female. But then with monoecious, we can break this down even further. You can have sequential hermaphrodites, which are animals that start out as one sex and then change to the other later in life. Or you can have simultaneous hermaphrodites, which is where from the get-go, each individual is both male and female at the same time. Then there's also this concept of self-fertilization at the bottom. So some sexual reproduction species are able to self-fertilize, where 2 of an organism's own gametes fuse. Just take note of the fact that this is different from asexual reproduction. It still involves combination of gametes, still involves fertilization. It's a type of sexual reproduction, not asexual. Then moving on to the right side, we have asexual categories. So there's obligately asexual, which just means a species can only reproduce via asexual reproduction. And then you have facultatively sexual or asexual, where they can use either mode of reproduction. So facultatively sexual and facultatively asexual are both names for the same thing. Next slide. Okay, so the size advantage model of sex. This model is used when we're looking at sequential hermaphrodites, animals that start out as one sex and then change to another later in life. We use it to explain what sex the animal will start out as. Will they start out as male first and become female later in life? Or will they start out as female first and become male later in life? So animals will change sex according to when it's most advantageous. So if we look at this chart on the right here, this is looking at clownfish. On the y-axis, we see reproductive success, and on the x-axis, we see size. Size is just going to increase as we age, so the x-axis is also just kind of age as well. Then we see two lines, a blue line representing males and a red line representing females. What we see is that on the left side of the chart, when clownfish are small and when they're young, males will have greater reproductive success than females, since the blue line is above the red. But then later in life, once the animals have grown and they're older, we find that females will have higher reproductive success than males. So what we find is that because it's most optimal to be a male early in life when you're small, and most optimal would be a female later in life when you're large, we find that clownfish will start out as male and then become females later in life, as that's what maximizes their reproductive success as they age. So can you just do one transition? Yeah, so that previous chart was for clownfish, clownfish will start out as male and become female later in life. But this isn't the case for every animal. It's going to be different. With blue streak wrasse, it's advantageous for them to be female when they're small and then become male when they're larger. So it's going to vary by animal. Then lastly, just two terms to know about this is that protandry just refers to when the hermaphrodites start out as female and as male and become female later in life, and protogeny is starting out as female and becoming male later in life. Okay, next slide. So we're going to go over some advantages of sexual reproduction, starting with Muller's ratchet. So Muller's ratchet talks about how asexual populations will increase in their number of harmful mutations across each generation. So within each asexual individual's life, they're going to accumulate mutations through normal means. These could be DNA replication errors, radiation, anything like that. They're going to accumulate mutations over their life, and then when they reproduce, they're going to clone themselves and pass down, along with their entire genome, they're going to also pass down these mutations. Then the same thing is going to happen with the offspring. They're going to accumulate more mutations, pass them down, blah, 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 keeps continuing. So with asexual populations, there's no way to get rid of these accumulated mutations. What sexual reproduction does is it adds a way to remove these mutations. So due to meiosis in sexual reproduction, we only take a portion of each parent's DNA, half from your mom, half from your dad. Whenever this happens, there's a chance that each gamete will only take the DNA that doesn't have any of these mutations. So they won't inherit these accumulated mutations from each parent. Of course, this can also go the opposite way, where each gamete might only inherit those mutations, might inherit all of them, but there's a chance that they won't. So if you have multiple offspring, what you'll find is that some of them will have these mutations removed, and some of them will inherit all of them. The ones that had them removed are probably the ones that are going to survive and keep going forward. But yeah, that's what the advantage of sexual reproduction does. It allows for a way to remove these accumulated mutations. Then, so that idea, Muller's ratchet refers to the idea that the number of mutations is just going to keep increasing with asexual populations because ratchets only go in one direction, just like how it'll only increase. Next slide. Okay, so two more advantages to talk about. First is the lottery principle, which is kind of just repeating the concept of how sexual reproduction increases genetic diversity. This makes us able to adapt more to environments. But yeah, so the principle states that sexual reproduction should be observed more in unpredictable environments, and then you'll find asexual reproduction is observed more in stable environments that don't change very often. So as we talked about before, with sexual reproduction, you're increasing genetic diversity, and you're able to adapt to a variety of situations because each offspring will be different. Hence why you'd find them in unpredictable environments that are constantly changing. That's where you'd find more sexual reproduction. Then on the other hand, if you're in a stable environment where nothing's changing and the parent is able to survive just fine, then in theory, you should find that it's just asexual reproduction happening because there's nothing wrong with their genome. They can just pass it down to their offspring, no problem. But that's the entirety of what this is. But it is stated in lecture that there's not much support for this idea being the big reason for why sexual reproduction is so useful and so common with animals. Okay, that's all there is to know for lottery principle. It's called the lottery principle just because of that idea that some of your offspring will be, if you have a variety of offspring, some of them will be good and some of them won't be good. Yeah. Next is evolutionary arms races with other species. So we talked about how sexual reproduction can make offspring more fit than their parents. And if surrounding species are getting more fit by using sexual reproduction and you aren't, you are going to fall behind, and you need to stay ahead of the curve. If all the animals around you are using sexual reproduction, getting more fit, evolving, and you're just staying there using asexual reproduction, and your fitness is going to fall behind all your other competitors in your environment. Okay, that's what the big idea is here. Then there's a lot of emphasis placed on this for when you're looking at parasite interactions, because using sexual reproduction, you're going to keep changing your genome. Parasites won't be able to adapt to your body type. So with sexual reproduction, your body type's going to keep changing. And this whole idea, not specifically with parasites, but the whole idea of just staying ahead of the curve is known as the Red Queen's principle, which is something you have to know. Next slide. Okay, so disadvantages of sexual reproduction, and there's just three. First is the cost of mating. Sexual reproduction takes time, takes resources. That's it. Next is cost of males. So if we were in a population that only had females that reproduced via asexual reproduction, what we find is that 100% of the population is contributing to adding more offspring. But with humans, we instead use sexual reproduction and have males and females. What we instead find is that now only 50% of the population is actively contributing to adding new offspring, this being all the females. So what this means is that by having this sexual reproduction and this concept of male-females, where males don't actually actively produce more offspring, it means that our populations grow slower. That's why cost of males is a disadvantage, because it makes our populations grow slower, since only half the population is actually adding new offspring. Then last disadvantage is the cost of meiosis. So just in general, a big motive is that we want to survive and pass down our genes. But with sexual reproduction, you're making it so that you're only passing down half of your genes and the rest gets diluted by someone else's, which is why we say this is a disadvantage. Next slide. Okay, so sexual selection, just another term to know. Sexual selection favors traits that increase mating success, even if they reduce survivorship. Obvious example is that male birds are often bright and colorful because the females find that attractive, even if that gives them a lower survival advantage and makes them more prone to predators. So sexual selection means selection referring to being able that improves mating success as opposed to improving just survival. Then two more terms, kind of just memorization. Intra-sexual selection refers to male against male competition for female mates, whereas intersexual selection just refers to the idea of just females choosing and the males just try to be the most attractive version of themselves possible. Next slide. Okay, so just the concept about how anisogamy ties into like male and female differences. So anisogamy, which was the idea that gametes can are either going to be small and mobile or large and non-mobile, basically just means sperm versus eggs. So anisogamy puts different selective pressures on males and females. This is because sperm is cheap to produce and eggs are expensive. So what we find is that the selective pressure with males is that they want to produce the greatest amount of offspring and attract as many mates as possible, maximizing the quantity of offspring. How anisogamy affects females instead is that because gametes are more limited and costly, they focus more on the quality of offspring, hence why they're more choosy. So this is why you might hear that anisogamy is the reason that females will be choosy. Okay, next slide. Okay, so the mating game in lecture, I'm just going to quickly summarize it. So there's nine females with a few gametes, nine males with lots of gametes. Everyone receives a quality rating from 1 to 5. Mating happens, and at the end of the game, the person with the most points based on the number of offspring and based on the offspring's quality ratings will win. So there's only 4 things you need to take away from this entire part of the lecture, which is that first, the highest quality male wins the mating game. The lowest quality male loses the mating game. Males and females share the same average fitness, and this is just because of the fact that there's an equal number of males and females. That's why this fact is true. And then lastly, males have a higher spread in fitness and a higher potential fitness too. This fact isn't super, they don't elaborate on it too much in lecture, but it does intuitively make more sense if you consider the fact that females are choosy, which gives males a reason to want to compete to be better and better, and for them to want to have a higher potential fitness. That's why their fitness varies more. Next slide. All right, so what is the benefit of or of one sex being choosy with the other? There's three different terms to know. So first is direct benefits. So attractive mates make good parents and would provide a tangible benefit like food, territory, or protection. So this generally this is when we're talking about both sexes being involved in parental care. Next is indirect benefits, which is the idea that attractive mates have good genes. So something about the males might make them appear attractive or fit. Like that, for example, in lecture, she gives the example that a frog might sing very well. Whatever trait this is, it indicates that they have good genes. So the idea is that even though the male might not be involved in parental care, there's still an advantage to mating with them because they have good genes. The good genes will help the offspring thrive and survive. That's all. That's what indirect benefits is about. Then lastly is a taste for the beautiful, or also known as the sexy son hypothesis, which has to do with attractive mates produce attractive offspring. So there might not be any rational reason for why a female considers a male attractive. In the lecture, she gives the example that a frog might be bright orange, which doesn't really provide any survival advantage, but the females will find it attractive regardless. The idea here is that it's still advantageous to the female because if they mate with this bright orange male, their sons, the female's sons, will also be bright orange. And the sons, because they're bright orange, will have an easier time mating and finding females and making more children. So in the end, mating with the bright orange male that all the females consider attractive increases the female's chances of having grandchildren and spreading her genes down the line. That's what the benefit is. Next slide. Okay, and then lastly, which sex will be choosy? So this is easy. Whichever sex is more invested in parental care will be more choosy. If both of them are equally invested, then both of them are going to be choosy. That's it. Next slide. Okay, so first multiple choice question. You guys can pause the video because I'm going to go over the answer now. So next slide. Okay, so I'm just going to go through each answer one by one. Males are more likely to compete with each other for female mates due to high gamete production. This is not going to be true because the males are invested in parental care, are the ones most invested in parental care. As we just talked about, whichever sex is more involved in parental care is the one that's going to be choosy. Since the males here are going to be choosy, they're not going to be competing with each other. We'd find that females should be competing with each other for males. Then next with B, both sexes will avoid choosiness since both share equal parental responsibility. That just goes against what the question said because the male has more parental responsibility. C is true. Intrasexual selection involving competition for male mates. We found that the females would, because the males are invested in parental care, it is feasible that we find intrasexual selection where females compete for male mates. It's not necessarily going to be intrasexual selection, but it is a possibility, and that's why this is the correct answer to the question. And D is females will continue to be the choosy sex regardless of the level of male parental care. Just not true. It just has to do with amount of how involved they are in parental care. Next slide. So you guys can read over this. I'm going to go through each answer now. So next slide. Oh, yeah. So the first one is male individuals typically have greater fitness than females. This is not true. What is true is that male individuals typically have a greater spread in fitness than females. So they might have a greater potential fitness than females. But on average, male and female fitness is equal. The averages are equal. Next is individual clownfish start out as male and become female later in life, making them an example of sequential dioecious species. It would be correct if it said sequential monoecious species, but it says dioecious, so it's wrong. Monoecious means both male and female. Dioecious means just one of the two sexes, like humans. Then next is the Red Queen's principle suggests that sexual reproduction is useful in increasing genetic diversity of offspring. That second half is true. Sexual reproduction is useful in increasing genetic diversity, but that's just not what the Red Queen's principle is talking about at all. Red Queen's principle has to do with the idea that it's advantageous to have sex, to use sexual reproduction, because keeps you ahead of other animals. That's all. That's what it's about. Then lastly, D, a hermaphroditic species in which male individuals have greater reproductive success when they are small in size likely demonstrates protandry. This is actually just talking about the exact same situation as the clownfish. Hermaphroditic species are ones that are both male and female. If the males have greater reproductive success when they're small in size, it means the animal should start out as male, which is what protandry is. And I think next is cycle 8.
Okay, hi everyone. Thanks so much for your patience. I know we're almost there. So, cycle 8 is relatively short, so we'll just try to get through it as quick as possible. So, in cycle 8, we really build on species and speciation. And what we define really in class is that biologists have different ways to define a species. So, the three main ones we go over in the course are morphological, biological, and phylogenetic. So when we look at the morphological species concept, this one's relatively simple. So it tells us very little about the evolutionary processes that produce a new species, but it basically is like if two different organisms look the same and share similar physical traits, then they are the same species. So it doesn't really distinguish species that look identical but actually are different, and it just essentially assumes that individuals that look different are separate species. So for example, based on the morphological species concept, humans, if you're a male or a female, would be different species because you don't look the same. When we look at the biological species concept, this one pertains a little bit more to if members of a species can interbreed and produce a fertile offspring with each other, then they are the same species. So basically, if you can reproduce to create an offspring, then you are the same species. But the problem is that this doesn't really account for asexual organisms, so any organisms that kind of just produce without needing sexual reproduction. It doesn't apply to shy organisms, so essentially one species that we would have a very difficult time observing if they do reproduce. And it doesn't apply to extinct organisms just because we can't actually see anymore if they can reproduce with each other because they don't exist. And finally, we look at the phylogenetic species concept, and this one is basically looking at a phylogenetic tree. So you just take a small, essentially, branch or small, like twigs of the same branch, and you group them all together and say that they are the same species. So it uses morphological and genetic sequence information to construct an evolutionary tree for organisms. The problem with this one is that you can't really use gene flow. So with a phylogenetic tree, you can't actually see which individuals are able to breed with one another. It's kind of just like clustering them together. So obviously you can see with these three different concepts, there's obviously pros and cons associated with both. So especially for the exam, you want to be pretty specific when you're reading the questions. So they might give you a description of two different organisms and say, give you some characteristics of how they interact and say, well, based on this description, like what concept would these two organisms be considered the same species or different species? So just being able to distinguish between these three is really important. So we'll go to the next slide. And the next thing we want to talk about are isolating mechanisms. So isolating mechanisms really relate to if two individuals or two organisms are able to produce an offspring and probably why they cannot. So we define these different types of mechanisms in class as either prezygotic, so either before the zygote, or postzygotic, so after the zygote. And the zygote is essentially what forms between the sperm and the egg. So when we look at prezygotic isolating mechanisms, this basically means any mechanism that prevents the egg from even like fusing with the sperm, so they'll never ever create a zygote. So the ones that we go over in class are ecological, meaning that these two organisms kind of just reproduce in completely different areas, so they'll never really meet. Temporal, meaning that they produce at kind of different times of the year. Behavioral, meaning that they just have very different reproductive behaviors, so they would not really recognize each other. Mechanical, meaning that these two organisms cannot even physically interact to reproduce. Or gametic, meaning that these egg and the sperm is completely incompatible and they'll never fuse together. When we look at post-zygotic isolating mechanisms, this occurs after the zygote forms. So the sperm and the egg are able to fuse together, but the offspring is either inviable, sterile, or we have something called a hybrid breakdown. So inviability really relates to the fact that these two, the egg and the sperm that fuse together from these different organisms, might have very different and distinct gene/developmental instructions per se that just basically do not allow a offspring to form. So fertilization does occur, but the organism will die young or it does not even survive to, I guess, like maturity. Sterility really relates to hybrid can grow up but becomes unable to produce like more functional gametes. So you basically just produce an offspring, but the kind of the familial line will end there. And finally, hybrid breakdown is probably maybe the most confusing one, but if we break it down a little bit easier, it's really relating to the fact that a hybrid can develop, so you will have an organism that develops, but they will be able to mate and they will be able to produce more offspring. But the problem with these offspring is that the fatality rate increases and the fertility rate decreases. So as these kind of hybrids make more offspring, the offspring will die more quickly and it'll be more difficult to produce more offspring as the generations go on. So essentially, it basically reduces fitness down to zero. And when you have that, you can't really create a new species because your species will die off pretty quickly. So that's basically the recap of post and pre-zygotic isolating mechanisms. And we can go to the next slide to talk a little bit more about speciation. So speciation occurs mainly through two mechanisms, which you learn throughout this cycle is either allopatric or sympatric. So allopatric speciation refers to when basically a group of organisms becomes geographically isolated. So it's either maybe like a river or a highway that separates, like physically separates this group. So like you have a group of like wolves per se, and then all of a sudden you like build a highway right through. Over time, these two, I guess, distinct groups might diverge in like their gene flow. So essentially they'll be able to accumulate different mutations and they'll have genetic differences to the point that they may not even they may not be able to come back together to reproduce with each other again, even if they like were initially the same species. The second one is sympatric speciation, which I guess is a little bit confusing to think about because conceptually it is a bit odd, but essentially it's looking at if you have a group of organisms and somehow within that group, with no geographic isolation, you'll have a new species form. And the main example that we really talk about through this course is the polyploidy in plants, because typically with, I guess, more like mammalian type of reproduction, it's a bit slower, so this wouldn't really occur. But with plants, what can happen is that you can have something called allopolyploidy, which is when the DNA from two different parent species replicates, but the cell doesn't divide. So you'll actually have chromosome doubling. And the gametes can fuse with each other, but not with the gametes from the original parent population. So basically, just because with the imbalance of chromosomes, you might not be able to kind of reproduce, the offspring might not be able to reproduce back with the parent. So then those gametes, the ones with like, I guess, the doubled amount of chromosomes, will now only be able to fuse with each other. And then this will create kind of a new species within like the same area or same population without the geographic isolation. And this really pertains well to the next slide, which we will talk about secondary contact. So with secondary contact, what occurs yes. With secondary, what contact what occurs is basically when you have two previously geographic isolated populations and they come back together. So if you're looking at allopatric speciation, let's say those two groups of wolves that were separated by the highway are able to kind of interact again, then that is what we call secondary contact. And there's two main kind of, I guess, results of secondary contact. One, you can have reinforcement of speciation. So it actually increases the rate of speciation because if reproduction isolation occurs and these two groups are no longer able to mate with each other and produce offspring, then you won't have a new species form, or I guess like respecies. You won't have that I guess reproduction. So therefore, speciation will actually be sped up. or you can have resume interbreed, or you can just resume interbreeding, which essentially will slow down the rate of speciation. So if populations are able to successfully interbreed, they can essentially just fuse back together, gene flow can continue, and then they become one species once again. So that's really the recap of this cycle. So we'll go over two questions to kind of make sure that you really can solidify this. So we'll give you guys a second, pause the video, and then we'll go over the answer. So if you read the question, Hannah, you can go to the next slide. Yeah, you'll see that the answer is B. So we'll go over this answer pretty quickly. Basically, when we'll just go through why A is wrong and C is wrong. So A is wrong because the words in the passage specifically relate to that if male bowerbirds do not provide any care, they do not provide any care at all to their offspring, which indicates that there's actually no direct benefits provided, meaning that A would be wrong because it's saying that those males would provide the best direct benefits. And if we look at C, it says the scenario within the paragraph, it says that the females prefer to mate with males who build attractive bowers and not mating with the attractive males, essentially to produce those sexy sons. So what we're looking at this is that we have to be really, really specific with the wording. And B is correct because in the passage, it says that females mate with males that are good builders and basically means that they have high quality genes and improve the health of their offspring. So just making sure that you guys are really specific when you read if it says like direct benefits, indirect benefits, those are typically what trip people up on the exam. So just making sure you're highlighting those words specifically. And also D is just saying A, B, and C probably contribute to why females prefer males. You just also want to say like if you are not really sure if it's A, then it's probably not D. So it's just kind of a process of elimination as well. And we'll go over one more question with question 2. And this is looking at allopatric speciation. So you can pause the video here, read over the question, and we will go over the answer. And we can click to the next slide. And so the correct answer is C. So we'll go over quickly why A, B, and D are wrong. So with A, this one is really specific to the wording because it's saying as soon as the populations become physically isolated, which is incorrect because immediately after separation, these populations would still be able to form a zygote. Because when you look at evolution, that leads to two populations acquiring like different genetic differences. It does not happen within a generation or immediately. You need quite a bit of time to pass. So that is why A is wrong. So being really specific with that as soon as wording. When we're looking at B, this is incorrect because prezygotic isolating mechanisms are favored upon secondary contact. And this one's saying before populations even come into contact with each other. So without a reunion between these two populations, there will be no attempt at fertilizing or gene flow. So thus, like prezygotic mechanisms do not even apply to this scenario. And when we're looking at D, this is incorrect because if two populations become post-zygotically isolated, then they've already overcome the pre-zygotic isolating mechanisms because your post mechanisms become after your pre. Essentially, what this is saying is like if you already are looking at post-zygotic isolation, then the pre-zygotic isn't even an issue, and we're only looking at the ones that are after the zygote formation. But that's the recap of cycle 8. I will pass it off to cycle 9, and I think that this one's a relatively simple cycle, so just making sure you guys get all your terminology down is the most important thing.
Okay, thanks, Abby. Hi, everyone. I'm Victoria. I'm going to go through cycle 9, evolutionary relationships. So the most important thing with the cycle is really just being able to analyze these evolutionary trees. You're going to see a lot of these. So with these trees, time moves from the base of the tree to the tips of the tree. And then just some other terminology. The root of the tree is the base, how you think of like an actual tree, the roots are the base. And then the nodes are the branching off points of the tree, and those represent speciation events, so the points in evolutionary time when there were kind of divergence in the lineages. So, and then one last term is taxon or plural taxa, and those are just the species at the tips of the tree, and that's kind of what we're going to look at and what traits those species have. So an important skill here is being able to recognize which groups of species are the most closely related to each other. So for example, if I were to ask you, are frogs more closely related to ducks or to sharks? What you would want to do is you would want to look at the most recent common ancestor or the merca of those groups. So for example, let's look at the frog and the duck common ancestor. If you trace back along the tree, the most recent common ancestor is going to be at that orange dot. On the other hand, if you look at frogs and sharks, their most recent common ancestor is going to be where the green dot is, which is further back in evolutionary time. So the group of species that has the merca that's further along, that's like more recent, is going to be the group that is more closely related to each other. So in this example, the frog and the duck are more closely related to each other than the frog and the shark. Next slide, please. So another important skill is being able to recognize groups that are monophyletic versus non-monophyletic. So a monophyletic grouping is a grouping that includes the most recent common ancestor of those species, as well as all of the descendants of that merca. Another way to think of that is that you only need to make one cut to the tree if you're drawing a circle around all of those species. So there's an animation here, Hannah, if you could just click forward one. Yeah, so if I'm looking at the frog, the duck, and the human, these species are monophyletic because their most recent common ancestor is right at that point in time where the scissors are cutting. And all of the descendants of that merca are included in this group. If I draw a circle around them, I only need to make one cut to the tree. So this is monophyletic. On the other hand, if you could just click forward one, the fish and the shark are not monophyletic because if I try to draw a circle around them, you see I have to make two cuts. Even if I try to kind of extend the circle to the bottom, like it doesn't work out. But another way you can think of it is what's the most recent common ancestor of those two species? It's going to be that ancestor that's at the very base of the tree. And this grouping does not include all of the descendants of that ancestor. So the fish and the shark are non-monophyletic. Another thing to note here is that many traditional groupings are actually non-monophyletic. And the reason why that is just because we named those groups and we came up with those groupings before we really understood the evolutionary history of those species. So for example, we often consider prokaryotes as one group, and that comprises bacteria and archaea, but bacteria and archaea are actually not monophyletic. Archaea are more closely related to eukaryotes than they are to bacteria. Next slide, please. Another important skill is visualizing trees. So you might be given two trees and asked, are these two trees the same? So try not to be fooled by like the orientation of the tree or the way that it's drawn. Really, you just want to look at which species are the most closely related to each other. So take a moment, look at these two trees. Are these two trees showing you the same evolutionary history? And if you could just click forward one, the animation will show you that yes, these two trees are actually the same. The reason for that is that we see that the flower is the most it's the most distant ancestor of all these species. It's kind of on the outside. And then we have the dog and the bird together, and then the mouse and the snake together. And no matter how it's oriented, you can flip around any node on the tree and it'll still be the same thing. Really just looking at which two species are kind of grouped together, which species are closely related to each other. And of note, this is not like an actual tree. I just made this up for illustration purposes. Next slide, please. So just some terminology, traits can either be ancestral or derived. So a trait that is ancestral evolved before the most recent common ancestor of that group of species. So you're thinking like ancestors, ancestral, before, whereas a derived trait is one that evolved after the most recent common ancestor of that whatever group of species you're looking at. And another kind of classification terminology that arises here are symplecomorphies, autapomorphies, and synapomorphies. So a symplecomorphy is a trait that is shared and ancestral. So shared among species, and the most recent common ancestor of that overall group had the trait. Autapomorphies are traits that are unique and derived. So only one species has it. That's a pretty easy way to recognize an autapomorphy. And then synapomorphies are traits that are shared and derived. So it evolved after the merca of that group, and several species have that trait. And these are going to be what's most helpful for us when we're building phylogenies. Next slide, please. So you might also be given a tree and you're going to be told, here's an outgroup, here's a trait. Is this trait ancestral or derived? So let's just go through 4 different examples. So in this example, the fish are the outgroup, the thing on the very left. And what if I tell you that a trait is present in the outgroup and in all of the in-group? Is that trait ancestral or derived? Think about it for a moment. If you said ancestral, then you are correct. This one's pretty straightforward. It's most likely that this common ancestor at the very base of the tree had that trait evolved kind of before all of these species, so it's ancestral. Next slide, please. So what if the trait is present in the outgroup and some of the in-group? Is it ancestral or derived? So the answer here is ancestral. And what we have to remember here is the principle of parsimony, which states that the tree with the fewest evolutionary changes is probably the most correct tree. So in other words, the simplest tree is probably the best one. The reason for this ancestral evolutionary change to get this tree is this ancestor at the very bottom had that, or sorry, the shark's losing the trait. If it's derived, we need 2 changes to get it. We need to have kind of the fish evolving that trait separately, and then the ducks, the frogs, and the human. So that's just too many changes. This is probably ancestral. Next slide, please. So if we have a trait that is absent in the outgroup and present in some of the in-group, what do we think, ancestral or derived? If you said derived, you're correct. That's the most straightforward explanation, probably evolved further along in time, somewhere between the sharks and the frogs. Next slide, please. And what about this tree where it's present in the outgroup and absent in the in-group? Do we think it's ancestral or derived? Or maybe it's both? The answer is both. So both are equally likely. The trait could be ancestral. There would be 1 evolutionary change required for that. So if the ancestor at the very bottom had the trait, so it's ancestral, we need 1 change here, which would be these 4 species kind of losing the trait in like along the bottom there. I can't point it out, but you kind of understand what I'm pointing to, which is 1 evolutionary change if that trait was ancestral. Or it could be derived. That would also require only 1 evolutionary change. Maybe that ancestor at the bottom of the tree didn't have the trait, but then the fish, which were the outgroup, kind of evolved to have that trait later on, then it would be a derived trait. So because both the ancestral and the derived explanations only require 1 evolutionary change, they are equally likely. Next slide, please. And then this is just some more terminology. A homology is a trait that is a similarity among species, and it reflects common ancestry. So that's a trait that's going to be helpful for us when we're building trees. But a homoplasy is a trait that is a similarity among species, but it's misleading to us because it actually doesn't reflect close common ancestry. So for example, bird and bat wings are an example of a homoplasy because birds and bats are not super closely related. They kind of evolved to have wings on their own, like in different lineages. And that's an example of convergence or convergent evolution. So just make sure you have this terminology down because it can be kind of confusing sometimes. Next slide, please. Okay, so now let's do a practice question, pause it, read it over, and I'll explain it to you now. So the correct answer here is B. Statements 1 and 3 are correct. So bears are more closely related to lizards than sunfish. We're going to look at the most recent common ancestors of these groups of species, and that's going to tell us the answer. So if we're looking at bears and lizards, their most recent common ancestor is the common amniote ancestor. But if we're looking at bears and sunfish, their most recent common ancestor is all the way kind of towards the bottom, which is the jawed vertebrate ancestor. So because the lizard bear merca is further along in evolutionary time, they're more closely related. Option 2 is incorrect because lamprey are equally related to sunfish and to chimpanzees. The lamprey sunfish common ancestor is the one at the very bottom, the common vertebrate ancestor, and so is the lamprey chimp ancestor. That's also their most recent common ancestor. So since the merca for those 2 groups is at the same point in time, they are equally related. Option 3 is also correct. It's the same kind of logic. So the newt lizard merca is at the same point in time as the newt bear merca. With option 4, that one's incorrect. Notice that we're looking at the same 3 species, but the wording is slightly different. So just make sure you're clear on like what we're comparing things to. So if we look at the lizard newt most recent common ancestor, that's going to be the common tetrapod ancestor, whereas the lizard bear common ancestor is the common amniote ancestor, which is further along in evolutionary time. It's more recent, so those 2 species are more closely related to each other. Option 4 is incorrect. Next slide, please. Okay, so just take a look at this table. Which of the following traits are synapomorphies? There's a hint here that pops up as an animation, which is that synapomorphies are traits that are shared and derived. So take a couple minutes, look this over. And the correct answer here is answer A. So if we look at blue color, that is a trait that is shared among species S, T, and U, and it's also derived because the outgroup or species Q was red. So that trait is derived. The most recent common ancestor of all of these species was red. It wasn't blue. Pink fur is a similar kind of logic. It's shared among species S and U. And it's derived because the outgroup and the most recent common ancestor of this entire group was brown, it wasn't pink. And then same again with spikes, shared amongst the no spikes trait is shared among species S, T, and U, and it evolved further along in evolutionary time because, as we can see, the outgroup did have spikes. Short height is not a synapomorphy because it is present in only one species, so that would be an autapomorphy. Okay, next slide, please. And then lastly, just try to build the tree. You might be given a table like this and asked where the species go. The tree is in the upper right corner for your reference. If it's helpful for you, you can also draw out where the evolutionary events happen, so where those traits evolved. And give yourself a couple minutes and then we'll go to the next slide and I'll kind of explain the tree to you. So when you're looking at that table, you just want to remember that the species that are the most similar to each other are going to be the most closely related to each other. So species S and U, if you were to look at that table, they have the exact same traits. So you already know that those are probably going to be very closely related to each other. And then if we go, sorry, Hannah, can you just go back one? If we look at species R, that is going to be the species that is the most distantly related because we can see that it's the most similar to the outgroup. So it's brown, it has spikes, it's the color red. And then it's just less similar to species S, T, and U. So we can automatically put species R kind of on the left there, right beside the outgroup. And then in terms of determining where species S and U go, again, species S and U are very similarly related to each other because they have the exact same traits. Species T is just one off. So species T is going to go on that third branch. If you kind of need to convince yourself why this tree works, just draw out multiple versions of the tree and draw out where those evolutionary changes happened. And the tree with the fewest amount of changes is going to be the tree that is the most correct tree. So if you try drawing out a bunch of trees, all of them, except for this one, are going to have some kind of configuration where they have more than 4 changes. This is the tree with the fewest amount of evolutionary changes, so it's the most correct. This is just kind of a skill that takes practice. So if you don't get it right away, that's okay. Just go back, do some practice questions. And yeah, I'll leave you off. The last slide is just a terminology slide. I'm not going to go through all of this, but this cycle just has a lot of vocab. Not super duper complicated. Just go back to the textbook and lectures if you need it. Hopefully, like if you know these terms, then that should hopefully get you points on the final. All right, thank you guys, and I will pass it off to cycle 10.
Thank you so much, Victoria. Hi, everyone. My name is Dorsa, and let me talk to you about Cycle 10, cooperation and conflict. So, as we first learned, the evolution of cooperation was kind of a big puzzle, and it was difficult to reconcile with natural selection, because why on earth would selection favor something that's self-sacrificing, right? However, we learn that although common, these behaviors aren't truly altruistic. But before we go over that, let's go over the possible social interactions that happen in nature. First of all, if there is a fitness benefit to both parties, namely the recipient and the actor, that is known as cooperation, and we can see this in cooperative hunting in wolves. Then, if there is a fitness benefit to the actor but a cost to the recipient, that's known as selfishness. And we see this when a new lion that's entering a pride kills all the cubs so that it could mate with the female lions. Then, if there is a cost to both parties, that's called spite. And this doesn't really happen in the natural world. Finally, if there is a benefit to the recipient, but an apparent cost to the actor, keyword being apparent because it's not actually a cost, this is known as altruism. So this could either be the sentry behavior of meerkats or squirrels, kind of standing guard and possibly risking their life in order to protect their other, friends in the burrow, or it could be reproductive altruism, where individuals aren't exactly risking their lives, but they are passing up opportunities to produce offspring. So this can happen in social insects, for example, wasps, bees, termites, ants, or in certain birds. For example, in lecture, you talked about the white-fronted bee eater, lovely species name. And so they have a territorial pair, and when they have offspring, those children don't actually leave the nest to have their own offspring. Instead, they kind of stay back and help and gather food and protect their siblings. So next slide. Thank you. Up until the 1960s, these behaviors were explained by a theory called group selection. It basically stated that individuals will sacrifice their own fitness for the good of the group. However, it wasn't strong enough to explain the persistence of altruistic behaviors because all it took was for one selfish individual to kind of mess up the system and cause harm to the entire group. This is known as the free rider problem, sorry. And so kin selection came along, and this helped us see that these behaviors aren't considered altruistic and are rather acts of cooperation. So we expanded our view of fitness from including only direct fitness, which has to do with the number of offspring that an individual has. And so this is really easy to make sense of in terms of parental care and why um, a parent would want to help their child because that's like directly their genes and their alleles being passed on to the next generation. But we expanded that to also include indirect fitness. So that's another way of ensuring your alleles are present in the next generation. And this time it's helping non-descendant, more distant relatives who have similar alleles to you to survive and have offspring. Altogether, this makes up inclusive fitness. And this theory was formalized by Hamilton's rule, which basically says that helpful behaviors are favored if benefits weighted by the degree of relatedness are greater than the costs. So if this is true, all in all, there is a benefit to the inclusive fitness of that individual, and it makes sense for them to help out, even though if at first it seems like they're causing a detriment to their own fitness. As you know, there is a better version of this formula, so we'll get to that in just a second. Next slide. So we don't always see eye to eye with our family members, and this is the case when we talk about cooperation because it inevitably comes with conflict. And this is because individuals have overlapping but not identical genetic interests with their relatives. In other words, there are asymmetries in the relatedness that cause disagreements over when helpful behavior is appropriate because every individual is focusing on themselves and what the benefit to their inclusive fitness will be. So Hamilton's rule was expanded to reflect this. It was expanded to become R1 times B is greater than R2 times C. And I won't read out what each term is representing. You could do that, but I will kind of give you a word version of the formula. So it's basically a helpful behavior is favored if the benefit to the recipient is greater than the cost of the helper. But keep in mind that all of the R terms are based on the focal individual's relatedness to the recipient if we're talking about R1 or the helper if we're talking about R2. So it's important to note that Hamilton's rule doesn't require the individual to actually sit there and do math. Like the bird isn't calculating, if I help this person out, what's the benefit going to be to me? That's not, even though that's pretty cool, it's not exactly happening. Instead, it favors instinctive behaviors and motivations and rules of thumb that we all kind of live with. For instance, we naturally care more for our immediate family than our more distant relatives. Humans, on the other hand, we could do math. So the next time I get asked for a favor by my little sister, you best bet I am pulling up Hamilton's rules. All right, next slide. So how do we explain helping behavior between unrelated individuals, though? We have to take a look at game theory, or more specifically, the bats or prisoner's dilemma. This is basically a game where two individuals or players have the choice of being nice or selfish to each other without knowing what the other person is going to do. And so there are outcomes from best, which is temptation, to worst, which is sucker's payoff. And this all these are all possible on this payoff matrix depending on what each individual does, whether they cheat or cooperate. If you play this game once, or if in a social interaction the two individuals are never going to see each other again, the rational choice is to be greedy, is to cheat, because regardless of what the other person does, you will get the better outcome. But typically in real life, we don't always know when our last interaction with someone is going to be. So, in simple terms, we act nice or we cooperate so that we receive potential benefits in the future. Next slide. Therefore, helping behavior between unrelated individuals is based on repeated interactions between them. This is also called reciprocal altruism. This isn't really intuitively named because it's actually not altruism, but a form of cooperation. And so the conditions for this to happen are that there needs to be small and stable social groups, and individuals need to have the cognitive ability to recognize, remember, and as a result, reward/punish helpful or non-helpful partners. And this leads to social scorekeeping and reputations, which is seen in a variety of different animals, for example, vampire bats, as talked about in the lecture, wolves, dolphins, and us. For example, last year when I was in first year, I would help my friends out with biology in the hopes that they would help me out with calculus, and they did, so that worked out. Next slide. Let's do a question. All right, I'm not going to read it out for you, but take a second, read it out, write out the formula, solve it, and when you're done, unpause. Next slide. All right, so the answer to this question is C. No benefit to your sister or your half-sister would make you incurring a cost worthwhile because your mother isn't related to your half-sibling at all. So it's not worthwhile for you to incur any sort of cost to your and by extension to her fitness by helping. Next slide. This is the exact same scenario. However, this time we're asking from your father's mother or paternal grandmother's point of view, when should you help? Pause it, take a while to see. Next slide. All right, so the answer to this one is A. It's important to note that your paternal grandmother is equally related to you as she is to your half-sibling. Therefore, those relatedness terms are going to be the same, and conveniently, they're going to cancel out of the equation, leaving B is greater than 10. So from your paternal grandmother's perspective, you should help anytime the benefit to your half-sibling is greater than 10, which kind of makes sense because the cost to you is 10 fitness units. So the second that becomes that cost is or the benefit is greater than the cost, you should help. Next. All right, so here is a relatedness chart with some helpful values. However, I want you to not only memorize this, like I don't want you to memorize it. I want you to go in and see that you could actually derive it so that if you're thrown some random, like distantly related family member, you're able to solve and find the relatedness. And just a general tip, when it comes to questions that kind of combine math and biology, it's always really good to write out the formula first and then translate the words to the terms in the formula. And yeah, that's it for me. I hope you have an amazing exam season, and I know you will all kill it. So thank you for listening.
Hello, so I'm Ida. I will be doing cycle 11, and this is your last cycle, which is great. You guys have made it very far. Once we're done this cycle, we'll go on to the lab content as well. But I'm just going to be covering lectures 22 and lectures 23. So on the next slide. So we're going to start with population growth models, and there are a couple of different like mini equations that you do need to know. The first one is per capita birth rate, and so per capita just means average per person, and this is equal to the number of births per year divided by the population size. And likewise, we also have the per capita death rate, which is equal to the number of deaths per year divided by population size. And so we can use both of these like mini equations to calculate the per capita growth rate or our R value. And so this is equal to the per capita birth rate minus the per capita death rate. And I've also added plus immigration and plus migration here because you can calculate growth rate by taking the number of individuals that are moving into a population and subtracting the number of individuals that are moving out of the population. And N is also just our population size value, which we'll just use when we're looking at the growth models. So our first growth model is the exponential growth model. And this is when resources are unlimited, conditions are ideal, and populations can just grow and grow and grow till forever. And so in this model, the growth rate is equal to our per capita growth rate, R, multiplied by the population size. And so this isn't nearly as realistic as real life. Real life, we are limited by resources. And so a more accurate growth model would be the logistic growth model. And so there's a certain population size that it can be sustained, and then that is called our carrying capacity or our K value. And so once populations reach this value, they cannot grow anymore. They've hit their limit. And so this logistic growth model can be calculated, the growth rate of it can be calculated by the equation R times N times K minus N over K. And so all this equation is saying is that as the population size is small, the growth rate will be kind of small until it hits its midway point. And then it starts to approach the carrying capacity. And so the growth rate will increase until it starts to approach that carrying capacity. And then that growth rate will kind of slow down because it's getting harder to sustain so many people. And then on the next slide, we have environmental factors affecting population growth. And so environmental factors can be split up into two different categories. We have density dependent and density independent factors. Density dependent factors are factors that their impact changes on population growth depending on how many of those individuals are close together or far apart. And so that includes things like competition for resources, crowding, parasitism, disease, and predation. So I have an example here actually. So if we click forward, let's say we have a whole lot of people just kind of jumbled up together. They're like hanging out, they're having a good time. And let's say a virus comes in. So that virus is going to go to town because so many people are really close together and that virus can spread really easily. And so now these people are all sick, which is just no good. And then if you compare this to, let's say you have a population of, even if it's just as many people, but they're all far apart, they're not as densely together, only a couple of individuals will get sick because they're not close enough to have that virus spread. And so that's kind of how density dependent factors kind of act. It's like it's dependent on how many people are close together. We also have density independent factors. And so that includes things like temperature, natural disturbances, weather, and quality of food. And so no matter how close together or far apart these individuals are from one another, it does not matter. Those factors are going to impact everyone the same way. It does not matter how close you are, they're going to impact everyone. And so I have another animation here. So yeah, let's say we have a tsunami. That's just going to wipe everyone out. It does not matter how close they are together or how far apart they are. That wave is coming. That natural disaster is going to come and affect everyone. And so the last point for environmental factors is that density dependent and independent factors can interact with one another to affect population growth. And so an example of this would be, let's say a heat wave comes in and it makes everyone really weak and kind of not, their immune system's not working very well. And so that increases the population's susceptibility for disease or infection. And so that's kind of an interplay between the two where they do kind of play off of each other and affect population growth together. So on the next slide, we have biological factors affecting population growth. And so these are also called intrinsic factors because they're dependent on the individuals themselves. And so first off, we have fecundity, which is a potential reproductive output. So individuals or species with high fecundity means that they have a higher potential or more potential to have a whole lot of offspring. And so this can be due to the age of reproductive maturity, whether that's sooner or later in their life. That can be due to the number of offspring per event. So, for example, humans have like one or maybe two, sometimes three offspring per birth, whereas some species like mice have like 10 in just one birth. And another factor that kind of contributes to fecundity is the number of reproductive events per lifetime. So, how many times are you giving birth? The next biological factor is generation time. And so this is the average time between two generations. And in general, larger organisms will have longer generation times, like longer times in between one generation and the next. So for example, for humans, it might be 25 years between your parents giving birth and then you giving birth, whereas for bacteria, it might be like 20 minutes. It's whatever it might be. So a lot shorter. And finally, we also have population age structures. And so these are the number of individuals per age group. And so what proportion of the population is capable of reproducing? And so we have population pyramids. And the big kind of takeaway of these pyramids that you might see is that the base, as if the base is bigger than the kind of older populations or older proportion of the population, that means that more people or more individuals as time goes on, will be a reproductive maturity. And so that means that more individuals will be able to give birth, and that population is just going to increase. And on the opposite side, if you have a base that is very small, that means that in time that the amount of individuals who are of reproductive maturity will be a lot lower, and so that population might actually shrink. And so all of these factors are linked to economic development. And so this graph at the bottom was in your textbook and also in lecture. And all this is saying is that as a population increases in technological advancements or healthcare, the kind of pressure to have so many children drops. And so death rate declines really quickly because you have all these advancements and people aren't dying as frequently. And a little bit later, birth rate will also start to decline. And so we'll see initially a large increase in growth rate and then eventually a decline because birth rate has declined to the point where it is below the death rate. Okay, and then on the next slide, we actually have a question. So, which of the following scenarios highlight a density-dependent environmental factor that affects population growth? Hopefully you have paused and come back. But the answer to this question, they are in the speaker notes as well, but the answer to this one is D, for only. So number one is wrong because all apples that are harvested this year that were rotten, that is quality of food. And quality of food is a density independent environmental factor. It does not matter how many people are close together or if they're far apart. All of those apples are rotten. No one can have those apples. No one's going to be able to eat those apples. And so quality of food is kind of like a tricky one because it seems like it should be a resource that everyone has to fight for. And so it seems like it'd be density dependent, but it actually is independent if it is the quality of food, not the quantity. Option number two is wrong because mice can have anywhere from 3 to 14 offspring. That's great, but that is a biological or an intrinsic factor dependent on the mouse itself. It's not dependent on how many people or other mice are around. That is an intrinsic factor, not environmental. Option number three is wrong because the drought is an environmental density independent factor. And so that drought's going to affect everyone the same way. Doesn't matter if you're close with everyone else or if you're kind of far apart, you're going to be experiencing a drought. And then finally, option number four is correct because this is a direct example of where planting crops too close together or too densely will affect the growth rate and it'll actually decrease the growth rate because not as many seeds can be produced. Okay, and then on to the next slide. So now we're on lecture 23. So this is a slide on how humans impact other species. And so in lecture, you were introduced to the IPAT framework, which just is impact. It just says that impact is equal to the size of a population, the affluence or times the affluence or the wealth of the population times the technology. And so a big thing that was stressed to you guys was that the wealth of a population has more of an impact than the population size. And that's just a really big thing to kind of know is that wealthier populations will have more of an impact even if their population size isn't as big. And it's obviously proportional, but that's just a big thing to know. And so humans impact species in a lot of different ways. We are predators, so we hunt, we fish, we like to kill animals. Well, some of us do. And we also modify the landscape. So this is kind of like building cities. And so this might destroy a lot of habitats, but for some organisms like pigeons that like to reside in cities, this would actually help them out or at least like kind of increase their possible living locations. We also like to cultivate and farm species for food. So like chickens or cows or pigs, that really boosts up their population because we're cultivating so many of them. Same kind of idea with agriculture. We're growing a lot of crops, and so that increases their population. But by doing so, we might get rid of a lot of forests. We might get rid of a lot of natural habitats and be affecting the species living there as well. We also do contribute to climate change a lot, and that kind of ties in with pollution. We might cause sea levels to rise, icebergs to melt. We might, acidify the ocean. And so just human activities in general can harm the environment and therefore affect the species that live in those environments. But sometimes we try to do good things. We have a lot of conservation efforts to try and save endangered species. And so that is a way that we kind of have a positive effect. And then finally, we have invasive species. So sometimes we introduce foreign species to a new land and they just kind of destroy the ecosystem. And if this happens, sometimes we also introduce biological control. And so biological control is just another species that is used to kind of control the population size of the invasive species. Okay, and the next slide. Okay, so now we have interspecific interactions. And so in that chart there, we have a whole bunch of different interactions that you are responsible for knowing. So we can go through a couple of examples for all of them. And so one example would be, oh my God. One example would be predation. So that would be when a lion hunts a gazelle. So great for the lion, not so great for the gazelle. We also have herbivory, which would be like a bunny eating some grass. Great for the bunny, not so great for the grass. We have parasitism, which is when an organism lives in or on a host and feeds off of it. And so that would be, again, great for the parasite, not so great for the host. We also have competition. And so I do want to highlight this one because it's not as intuitive as the other ones. Competition is negative for both species involved because they're fighting over a shared resource. And if you can imagine, it might be a lot easier to obtain that resource if those species didn't have to compete over it. And so both are kind of losing in that situation. We also have commensalism. And so an example of this would be, let's say it's a really, really hot day outside, the sun's beaming down, I'm really tired, and I see a lovely tree, and I go and I sit under that tree, and I'm getting some nice shade. I'm getting some nice relief from the heat. And so that's great for me. And the tree just does not care. I'm not affecting the tree in any way. So that's kind of a commensalistic relationship. And on the flip side, we also have amensalism, where one species is harmed and the other is unaffected. And so an example of this would be when you're a kid and you're like sitting in a field and you're kind of bored and you're just kind of picking at the grass, like with your hands, and you're just playing with it. That's actually an example of amensalism because you're killing the grass. You're not really doing it any favors. In fact, you're actually hurting it quite a lot. But the grass is not really doing anything for you. That action is not really benefiting you in any sort of way. You're just doing it because you're bored. And then finally, we have mutualism. And so this is a relationship where both species benefit. And so that would be, say, a hummingbird drinking nectar from a flower and then spreading its pollen everywhere. A specific type of interspecific interaction as well would be symbiosis. And so this is when one organism lives inside or on top of another. And so a really big kind of key thing is that symbiotic relationships can be mutualistic, so beneficial for both parties, parasitic or commensalistic. It just depends on the two species involved. Another kind of important note in interspecific interactions is that coevolution can occur. And so this is when species evolve over time to better interact with each other and improve their own fitness. And so a really key note for coevolution here is that both species must have a strong effect on one another, so positive or negative. So it does not include commensalistic relationships and it does not include amensalism, because both species have to have selective pressure placed upon them. They both either have to be trying to get as much out of the relationship as possible or try to protect themselves from the relationship as much as possible. Finally, we also have trophic levels. And so this is kind of like the food chain. So we have producers who are plants that like to make their own energy from the sun. And then we have primary consumers that eat the producers, secondary consumers that eat the primary consumers, and so on and so forth. So let's say in this diagram here, if there was an animal that ate hawks, that would be a quaternary consumer. And so multiple species can be part of multiple trophic levels at once. Like humans, for example, we eat producers, we eat plants, so we're primary consumers, but we also eat animals, so we might be secondary consumers as well. And then the final note here is that detritivores are separate. So these occupy their own trophic levels, and these are decomposers or scavengers. Okay. Okay, so we also finally have mass extinctions and the Anthropocene. So a mass extinction event is defined as when over 75% of the world's species go extinct in under 2 million years. And so we've had 5 mass extinctions so far. You are responsible for knowing them. So the first one occurred at the end of the Ordovician period, the second at the end of the Devonian period, third after the Permian, the fourth after the Triassic, and the fifth after the Cretaceous. And so if you want to just memorize them, you can. If you want a mnemonic, a good one is, oh, don't pretend to care, which is like called the apathy mnemonic. But a big question that has been brought about recently is if there will be a 6th mass extinction. So this kind of leads into the Anthropocene. And so the Anthropocene is a potential new geological period characterized by human activity. And so we say this may have started 500 to 70 years ago. Some people like to say that it started after the dropping of the first atomic bombs. And we do have geological evidence for this. And so that would be technofossils, which are pretty much just human artifacts, and then also radioisotopes, which are kind of just leftovers from radioactivity. And so we do have kind of proof that this is kind of occurring. And a big question as to why there may be a sixth mass extinction is because all this human activity has caused the rates of extinction to increase a lot more than we were originally projecting. And so the big answer and the big takeaway from your lectures of this is that we're not sure if there will be a sixth mass extinction, but hopefully not. And so the big thing, again, another big takeaway of this is that all of the impacts that we have on other species, all of the negative things that we do, is not irreversible. It is not confirmed that we'll have a sixth mass extinction, and every bad thing that we're doing will not just continue to go on and things won't go bad. As your prof said, if you kind of take in a One Health mindset, a more holistic viewpoint for human activities, there are chances to kind of save yourself from the dystopian future, as she calls it. And so if you want to take anything from the Anthropocene lectures, it's that human activities do bad things sometimes, but we can save it still. Okay, and then next slide. So we have a question here. So which of the following interactions could result in coevolution? And so if you've paused it and if you've come back, the answer is actually C, 2 and 4. And so as I stressed before, coevolution occurs when in any relationship, or it can occur in any relationship that isn't commensalistic and isn't amensalistic. So option 1 is when a cow tramples on grass. This is an amensalistic relationship. The cows don't really benefit from it, but the grass is very much hurt. And so the cows have no selective pressure placed on them, so no evolution would occur here. Second, the influence of viruses of infecting human hosts. This is an example of antigenic drift that was actually brought up in lecture. And so the viruses that have antigens that can evade a human's immune system will go on to survive and reproduce, whereas human hosts that have strong immune systems can survive and also reproduce. So it's kind of this arms race as well. Option number 3 is an example of commensalism. The jackals do gain from the tigers, but the tigers have already moved on. They do not care. They're not really involved anymore. And so there's no selective pressure placed on the tigers. And finally, we have foxes trying to catch rabbits. This is a predator-prey relationship. And if you can imagine, the fastest rabbits are the ones that will be able to evade the foxes and get away and kind of survive and reproduce, whereas the fastest foxes will be able to catch the rabbits and survive and reproduce. So it's this kind of, again, game of coevolution where there's selective pressure, and whichever ones get to benefit from the relationship most will survive. And then the next question. So which of the following statements is true? If you pause and then come back, the answer to this one is 4. So option number 1 is incorrect, because while we can take up multiple trophic levels as humans, we cannot be called producers. Producers are plants. They're the ones that can get energy from the sun. So we're not producers. We can't do that. Option number 2 is incorrect for a couple of different reasons. So this statement kind of assumes that the sixth mass extinction has already started, which we're not sure of that yet. It absolutely states that it started after the atomic bombs, whereas that's actually kind of just like an estimate and kind of something that scientists have thrown out there. And then finally, it kind of suggests that the rates of extinction increase was due solely to the bombs, whereas it's due to human activity in general. So it's false for a couple different reasons. And option 3 is false because, as we stated previously when talking about the IPAT framework, it is the wealth of a population that has more of an impact than the size of a population. 4 is correct because, as we discussed, the Anthropocene has kind of records and like physical evidence for it through technofossils and radioisotopes, because they might become permanent records in the Earth's geology. So that's it for me.
Hey everyone, my name is Trevor. I'm here to take you through the skill development sections. Just a note before we get started, big thank you to all of the presenters and the team members from BMP so far. This has been a marathon of a review session, and for the students out there watching, again, and as previously said, I would really suggest making sure to take some breaks throughout this session to really let yourself digest some of the information. But without further ado, let's get started with the skill development, and we'll start with Module 1. So if you could advance, thank you, Hannah. So this is talking about serial dilutions. This sounds like a hard concept, and it's something that historically has given a lot of people some trouble. But I'm going to try and break it down for you in a way that makes it a little bit easier to approach. So, first off, a dilution. This is a technique that we perform in science. It's performed every day in a variety of science labs in many disciplines, chemistry, biology, to name a few. Essentially, all you're doing when you're doing a dilution is you're adding some sort of solvent to a concentrated solution to make that solution less concentrated. When we talk about a serial dilution, that just means we're diluting multiple times consecutively. So you can see in the um, graphic on the left side of your screen, this is an example of a serial dilution scheme in which we are diluting the sample. We would say we're diluting 1 in 10. Now, what does that mean, 1 in 10? Well, when I say we're diluting a sample 1 in 10, we're talking about our dilution factor. And as you can see now on the right side of the screen, we are often asked to calculate the dilution factor for a specific dilution that we performed. So in this case, we'll use the first step as an example, and a really important formula that you've probably come across before, whether that's through high school chemistry or perhaps some of your other classes, for those of you in the sciences, and that is your standard C1 V1 equals C2 V2. Now, all this equation is telling you is that the concentration for your solution before you've made the dilution times its volume is equal to the concentration of the solution after you've made the dilution, again, times its volume. So in this case, we're solving for C2, and we're arbitrarily setting the concentration of C1 to be 1 in this example. So we're taking 1 milliliter, which is where you see the 1 milliliter in the numerator of the third line of the equation coming from, and we add 9 plus 1 to get the total volume, the total final volume of our diluted solution, 10 milliliters, giving us a concentration of 1 over 10. But recalling that because we set the concentration of the original solution to be 1, this is really equivalent to saying that we've taken the original concentration and multiplied it by 1 over 10. The short form that scientists often use to communicate this is by saying that the culture has been diluted 10x. And this can essentially be repeated because, again, that's where the power of a serial dilution really comes to play. By repeating these steps over and over again, we can actually end up with a final dilution of 1 in 10 to the power of 5 because we did it. If you can see, we've actually moved 1 milliliter 5 times in that schematic shown, which if we simplify goes to 1 in 100,000, which is what is written on the schematic. Now, if some of you are confused after that explanation, um, just try and practice it for yourself. Come up with different dilution schemes. But I want to show you, and if we could go to the next slide, I want to show you another common question that you can get with serial dilutions. So we're going to be using the exact same schematic that I just told you about and that we just went through. And this time we're going to be asked, and if you could advance to the next slide, we're going to be asked to determine the concentration of our culture in the original sample. Now, this is one of the rare cases in first year biology where you will have to memorize an equation, and that is the CFU per milliliter. So those are the units that we use to measure the concentration of a bacterial colony, for instance, or culture rather, is the number of colonies counted divided by the milliliters plated times the dilution factor. Now, it's important to note, and if we could go to the next slide, please. When counting colonies, we need to select a plate with between 20 and 200 colonies. Often when you have plates that have either very a lot of colonies, so much so that it's difficult to manually count them, or a plate that doesn't have enough colonies, which there's some questions as to how reliable that count is going to be. So just generally speaking, if you're given the options like you are in this schematic, we should be selecting the plate with 50 colonies because it's the only plate between this specified range. And so plugging those numbers that we get in, we have 50 colonies on that plate. We're plating a volume of zero point 0.1 milliliters, and that's indicated on the schematic as highlighted in the orange box. And our dilution factor, which is also indicated but on the schematic, is 1 in 10,000. And so completing the math, we get a value of 5 times 10 to the 6 CFU per milliliter, which is our answer in this case. So that is the concentration of the original colony shown all the way to the left on this schematic in the big beaker. And just important to note, for those of you who might be unfamiliar or need a refresher, CFU is a unit that we use to say colony forming unit. For this class, all you essentially need to know is that that's a unit for quantifying the amount of bacteria in a sample. And let's go to the next slide. Okay, moving to module 2 now, and I wanted to talk to you all a bit about primary versus secondary literature. Excuse me. So primary literature is when the authors conduct their own experiments, they use their own methods, and they come up with their own study, and then they present the results that they collected based on the data and the results they collected to an audience, and that usually takes the form of a journal article. Secondary literature, on the other hand, is when authors use very many, like a bunch, I'm talking in some cases hundreds of primary articles, and really look at all of the findings that different research groups are coming up with and attempt to synthesize this information. And usually this is done to kind of come to a consensus understanding or to reflect the diversity of scientific opinions within a specific field. Now, there are different parts of the article. I wouldn't necessarily say that you should memorize these. However, that is your choice if you do. However, I'd like to point out that a secondary article will likely not contain a materials and methods section, right? Like an author of a secondary article might write about so and so in their article used these methods. And it's interesting to note that these methods were different from these methods used in another article. But all of that discussion will be in the discussion section of that secondary article. It wouldn't make much sense for a secondary literature article to talk about the materials and methods for a bunch of experiments that the authors didn't really conduct themselves. Another important section that would probably only appear in a primary article would be the results section. So if you're seeing microscope images or graphs and figures, which include things like p-values and labels, then what you're probably looking at is a primary article, because that's again presenting data from data that those researchers collected themselves. And we can move to the next slide with that, keep things rolling. And let's just go through some terminology because there's a lot of different types of hypotheses talked about in this course, starting with a research hypothesis. So this simply explains a mechanism and it is not directional. There's an example relating to wolffish written on the slide, but this is different than a prediction, which offers a direction but no biological explanation as to why this might occur. So definitely important to note the differences between those two. It's also important to talk about statistical hypotheses, which we can actually go to the next slide because this information is repeated. But essentially, a null hypothesis is a statement of no difference. Put in simple terms, if I'm comparing group A and group B, my null hypothesis is that there's no difference between group A and group B. My alternative hypothesis So you'll see this denoted by H subscript A. That's your statement of difference. That's my statement saying, okay, group A is different than group B. It might be higher, it might be lower, but we know that group A is different than group B. And you might be wondering, why do we use these statistical hypotheses? What's the point? Well, in science, we like to be very strict about when we say things, and we have a very high burden of proof. So we need to collect evidence that supports the alternative hypothesis. But this means that if the evidence isn't strong enough or it doesn't hold up under scrutiny, then we're actually forced to accept the null hypothesis. This is almost, it can be thought of as the default result. If we don't do a very good job of collecting high quality data, then we're going to have to accept the null hypothesis. And so this would be, again, that case where they are not significantly different, two groups. However, only when the p-value exceeds the critical value, and this is determined by tables, are we able to reject the null hypothesis, meaning that the alternative hypothesis is then supported. So that is when our evidence is high quality and it does support there being a difference between the two groups. Now, it's hard for us to predict the questions. We're students just like you. So I know a lot of you have been asking, is this something I will be expected to do on an exam? And I will say it is possible. However, if you are expected to do a chi-square analysis on the exam, you will be given a table, likely a very simplified version of a following chi-square table. If we could go to the next slide. Yeah, so this is what a chi-square table looks like. The main thing here to note is that, again, and if we could just go back one, Hannah, the degrees of freedom is always n minus 1, where n is the sample size. And then based on that, we can find, based on the table, the appropriate critical value to use when conducting the chi-square test. We can go to the next slide now. So what is a p-value? Well, a p-value is explained in a lot of ways. It's often explained wrong, believe it or not, and there's actually a lot of misunderstanding as to what a p-value actually is. In simple terms, a p-value is the chance or likelihood of observing the difference that we observed, so of getting the data we collected, when comparing two groups, assuming that no actual difference exists, right? So if we're assuming two groups are the same and there's no difference between them, but our data says this group is way higher, this group is way lower in any sort of measure you want to think about, then the chances of that happening randomly are very, very low. And that's why we have a low p-value. That, is just one way to think about p-values. Definitely something helpful to take into the exam. Let's go to the next module, population genetics in action. So it's my understanding that you actually were able to perform this experiment in your labs, which is a very cool experiment to perform. Essentially, you were taking advantage of the fact that this enzyme, the MDH2 enzyme, is present in 3 different forms or alloenzymes within the population you were studying. And the fact that they were present in different forms means that when run on a gel, their banding patterns that you will observe will be very different. So I've summarized the banding patterns in this figure, I believe is straight from your lab materials. So definitely something to review for the final exam. Make sure you know and understand these banding patterns and the information that they communicate. We can go to the next slide. Right. Okay. Perfect. So for this slide specifically, this is just an example of what you might see when actually run on a gel. It's also important to note that we've loaded these bands at the origin, which in the middle of the gel. So that might be different than some examples of gels that you see online because there are different ways to do it. But in this case, MDH2 is migrating towards the electrode that is negatively charged, which is the cathode. So a bit of confusing terminology there. Best to just review that and make sure it is straight. Okay, let's continue on. I know we've been, at this for a very long time. Oh, yes. Okay. We can go, I believe, one more. I believe there's some text. Yes. So, applying our knowledge from cycle 6 of Hardy-Weinberg equilibrium, we can actually use our observed data and you would have actually completed this in lab as well to calculate if the population were in HWE. So again, that's something you're going to know already for cycle 6. So just make sure and be aware that you could see it on the final exam. We can go next slide as well. Oh, yes. Okay. So module 4. So it is my understanding that you looked at some climate data and used some statistical software to practice analyzing large data sets, which is a very cool thing to get exposure to so early in your scientific careers. We can go to the next slide. I'd say for the exam, best to just brush up on and familiarize yourself with the type of data that this resource offers you in terms of climate data. And we can go next as well. Now, one of the big takeaways from this module that perhaps you observed in your session, perhaps you did not, but generally speaking, when you conduct a small study with class data, there can be some variability associated with that. And because of the small sample size, this data is actually less likely to be representative of the true difference that exists, or rather state of the population. In this, that's just a blanket statement, but I guess in this case, you would have been looking specifically at climate predictions and other measures. Whereas the larger your data set gets, actually, no, correction, you would have actually been looking at plant sizes, if I'm recalling correctly, like stem, different metrics measured with potting plants. Now, for the big field study, this is actually a data set that is more likely to be representative of the true population. And that just follows from the fact that they have a larger sample size. So variability plays less of a role in influencing those results. We can go next slide. If there is one, there is not. So with that, I will leave you with this for skill development specifically. Please review your ASA assignments. We've been told that, especially for the short answer question, that will appear from the skill development session, you're going to want to be familiar with all of the skills you would have developed, especially with those ASA assignments. So yes, I provided you a brief overview of the skill development modules, but in no way is that meant to be a comprehensive overview. Again, as I said earlier, it's difficult for us to predict the questions that will appear on the final exam. So better safe than sorry, and please remember to study all of your ASA assignments. Other than that, I want to thank you all for attending our final exam review. And again, thank you for being so understanding and flexible as we navigated a bit of the wintry weather that we received here in London, Ontario. On the screen are some resources for your access. So we have a BMP resource drive, which includes some exams from previous years. But if you come across a question that doesn't feel relevant to the course and what you were taught, please ignore it. The course content has changed over the years, and these exams will include questions that are no longer relevant to the course. As well, if you have any questions about a specific cycle, please feel free to join the BMP Discord. We have a channel for each cycle and a channel for lab content. So if you do have any questions stemming from this review session or just general inquiries, we will be happy to answer your questions on the BMP Discord. As well, we have a feedback form. So Hannah, I'm not sure if you wanted to quickly speak about the feedback form.
Sure. So it's just a quick form. You guys can fill out. It'll take less than 5 minutes. Just tell us a little bit about what you thought about this final review session. As well, if you have been to workshops in the past, it also asks you that. And we'll just ask you a couple of questions on any cycle workshops you have been to this semester. Yeah, 2 minutes. But yeah.
Perfect. Thank you, Hannah. On behalf of the Bio Mentorship Program, best of luck on your final exam for Bio 1001A. I know all of you guys are going to crush it and do super, super well. Thank you again for coming to our review session, even though it was asynchronous. And that's all for us for 2024, but we will be back for Bio 1002. So please look forward to that and keep on coming to the our workshops, our study sessions, and our enrichment events and sessions like this. So again, best of luck on the exam and thank you.